Hello, everyone. Welcome to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. Today, my name is Ken Hansen. I'm an associate professor in the Department of Chemistry and Biochemistry. I work with color chemistry and light matter interactions. But more importantly, joining me today is Professor Robb Tomko. He's a pharmacologist in the College of Medicine at FSU. Robb, if you wouldn't mind introducing yourself.
Sure. My name is Robb Tomko. I'm an assistant professor in the Department of Biomedical Sciences at the Florida State University College of Medicine. I'm interested in a bunch of things scientifically, but the major focus of my group has traditionally been understanding how cells get rid of or recycle proteins that they have inside that they either no longer need or become damaged in some way and need to be removed, much like taking out the trash. So as far as gaming goes, I got my Nintendo Entertainment System when I was about 6 years old, and I never looked back. I was talking to Ken a little bit before we started here. When in my prime, which admittedly was almost three decades ago now, I used to be able to beat the original Contra without dying. So I definitely don't think I'm going to pull that off here today, but I'm hoping to at least have a pretty decent speed run. Apparently, if I can top 23 minutes, that'll put me within the top 250 people in the world or something like that.
Yes.
Did I get that
right? Yep.
No
All
promises.
Right.
Don't get your hopes up.
The game for the day is Contra, 1988 release. Robb, if you wouldn't mind resetting, let's get started.
Sure thing. You guys see these high-powered 8-bit graphics? And,
quality.
I forget what it is.
Oh, yeah. What are you drinking today?
This would be Victory Iron Monkey, or I'm sorry, a Golden Monkey. This is one of my personal favorites, and it's from a small brewery in Pennsylvania where I grew up. So it's really potent. It's like almost 10% alcohol or something like that. So we should be having a merry... Oh, I'm already dead. We should be having a merry...
We're off to a good start. Yeah. I like it.
Anyway, I imagine as my motor function get worse and worse here, this will get really entertaining.
Well, it's okay. T-Man Gundy says you are the best farm professor out there.
Thank you. I do what I can.
Yeah. Well, now you got to prove your skills with gaming.
Yeah, you see that spread up there? I really would have liked to have had that, but that's okay.
Are we using cheat codes tonight? Yes, we are. We're going to be using Game Genie.
I would have to be competent in order not to use them, right? So, better safe than sorry. Although, I did a real quick playthrough a couple minutes ago, and I still remember a lot. So, I think this game will probably go pretty well. We actually have the entire Contra catalog, I guess that's a thing, queued up, so if/when I make it through this, we can try out some of the ones that I've never really played before, and that should be really entertaining. Oh, I'm already dead again. All right, do we have a death counter going just for fun?
If anyone in chat wants to keep track of deaths, feel free. You're at 1:20 time, but more importantly, does anyone have questions for pharmacologists / cancer biology researcher / drug discoverer Robb Tomko
/ rusty NES player?
That's okay. They're here for the science.
Yeah, definitely. There's probably plenty of good channels to go to if you want to see people do well at video games.
You know, I think,
hopefully this will be kind of like a train wreck where it's more fun to watch than, something going smoothly.
Calm Down Bronco asks, how much do you like Osmosis Jones?
You know what? I've never actually played that.
No, it's a movie, isn't it?
Is it? Oh, that's right. What am I thinking of?
Yeah. It's I think Chris Rock is the lead voice for the main actor.
That's right. I don't know that I've ever seen that. I am a fan of Osmosis, for what that's worth. It makes a lot of life possible.
Man, Osmosis Jones, that's a good question. I'd be curious how well they represent, I don't know, in an abstract sense, biochemistry and whatnot.
Yeah. I'm trying to remember what was the premise of that. It was like it all took place at like the molecular level or something. Or...
Yeah, and they had to within the body fight diseases and things like that. It has been a long time.
This is like the worst weapon to have for this.
So here's the thing, Bronco, is I'm going to go watch that sometime this week just to get a flashback of what
I
it's
know,
about.
I feel like I have homework now.
Oh, somebody asked me in a previous one if I'd seen the, what was it? Cloverfield Paradox, and I had not, so I had to go back and watch that one.
What was it called?
Cloverfield Paradox. It's the third Cloverfield movie.
Oh,
basically.
I saw that. It was not the best of the Cloverfields, I would say.
Yeah.
But there were, it gave me something to do was, I went the wrong way. See, this is what I was talking about. I'm wasting time right here waiting for this elevator to go up now.
Yeah, everyone has to spend that time. All right, so we'll keep pending on Osmosis Jones, and we'll get back to you after we watch the movie.
I expect someone to type a full synopsis into the chat box by the time I beat this.
Exactly. Scuzzbot will hold off on yours. What's your favorite class of drugs? Well, first, how do you classify drugs? Either as a whole, as a community, or not?
Yeah, so one of the more common ways to classify drugs is based on their target, the thing that they actually bind to in the cell in order to cause some desired biological change. The other way that drugs are sometimes described, and this is used a bit more commonly when the exact target inside the cell isn't known, is they'll be described based on what they are used for or what they do. So, like, there's a big group of drugs that are known as antihypertensives, for example, and they are used to lower your blood pressure. Hypertension is sort of the medical term for blood pressure. So, when many of these were discovered, they didn't actually know exactly how they worked. They just knew that they had this chemical that when they gave it to people, their blood pressure would go down. So they called them antihypertensives. And it wasn't until much later in our sort of scientific evolution here that people started to figure out how they work. So nowadays, having a drug that works but not knowing what the target is far, far less common. So most new drugs nowadays are named based on what their target is, or they're classified based on what the target is.
So of those, what is your favorite class?
What is my favorite class?
The ones that work?
Well, yeah, the ones that, treat conditions and don't make people sick, I think are good. I am trained as a cancer pharmacologist originally, so my default is to go to that. But then, that's like, I've never taken any of them, and nobody ever takes them under like relatively positive pretenses. So I don't think that's a good class to go with. We could go with, hallucinogens or something fun, right? I'm not claiming to have taken them, just to be clear, but
yeah, hallucinogens are a good class. Well, no, yeah. They're good for therapies now, right? Like PTSD.
Yeah, actually, so an emerging function or an emerging use for a lot of them is in PTSD and treating certain forms of depression. So they appear to sort of recalibrate the brain with respect to what is a good stimulus versus a bad stimulus, which is kind of cool.
Do you know how that works? Is that just an overwhelming reset, or how does it?
Yeah, so I think the short version is people are still trying to figure this out, but there are certain things there's certain parts of the brain that are associated that are responsible for storing, reward or anti-reward with certain types of experiences, and that sort of becomes uncalibrated in people who have depression or PTSD. So the basic idea is that by taking these drugs, you sort of dissociate whatever relationship is there, and you can reprogram starting fresh, good versus bad. Drink break. What's truly sort of a positive versus never. I am not the best neuropharmacologist, so I don't want to speak too deeply on this. Yeah, but
no
that's
worries.
Yeah.
Man, I had a profound moment in grad school talking about drug classes. Sorry, you had more?
What's that?
I had a profound moment in grad school when I was I was talking with one of my friend's wives who was a biochemistry / chemical engineering type person, and she was working on drug discovery. And she and I were having a conversation, and I asked her what kind of drug was it, and she started talking about the mechanism. In my brain, I'm like, is it a heterocycle? Is it, is it steroidal? Like, I'm a chemist. I'm all about structure, but that doesn't.
Kiddo, you got to come out. Sorry,
That doesn't
my kids are
apply
debating.
Oh, here's an important question.
Good night, kiddo.
Any extra credit for the quiz on Thursday?
Yeah, you guys keep pushing for that. I can't make any promises, but you never know. I do throw in extra credit from time to time, but I usually try to at least make it derived from the classwork rather than my extracurricular activities for a variety of reasons.
There is a follow-up, please, if that changes anything.
Well, I mean, that's polite for sure, so I respect that. And I would say if I decide to do so, I'll be slightly more inclined now with that, please.
It's good.
These bubbles are killing me.
Get on his good side. All right. Slightly more serious follow-up. What has been your most memorable moment in research or teaching?
Most memorable.
Oh, that could be positive or negative.
Yeah. So, I think for me, I have certainly had my share of highs and lows, just like any scientist. Science is kind of feast or famine, right? Things are either going really well or they're not working at all. At least in the biological sciences, I think this is the modus operandi. So, it's been highs and lows. There have been a few times, I think, where, I had discovered something, or at least I thought I had discovered something really important or really interesting, at least to me, and hopefully at least one other human on Earth. And I needed sort of like one incredibly difficult thing to shake out in order to, oh man, I'm blowing it here, to sort of seal the deal. So I can give you a good example of this. I published a paper, I guess it would have been like 2014, so it's like 6-7 years ago now. This is when I was a postdoc. And, I had all these pieces lined up to tell how this one protein that no one understood was functioning. It's basically kind of like an assembly chaperone to help put the proteasome together. The proteasome is sort of this very large garbage disposal inside each of your cells that will chew up proteins back into amino acids, which are kind of like the building blocks that proteins are made out of. And one of the things I was really interested in is how this thing was built. It's kind of like a car. It has lots and lots of different parts. There's maybe 70, 75 different parts that have to be put together. And I don't know if any of you have ever taken, a car apart or anything that's sort of very carefully engineered, but usually there's an order in which you have to do it to make sure everything fits together properly, right? So this was certainly the case for the proteasome, and I had been trying to figure out how this one thing worked. And in order to sort of convince myself and other people that I was right, I needed to basically use a pair of molecular scissors to cut this protein in half and show that it messed up this process. So, we didn't really know what this protein looks like. So I was just kind of taking guesses on where we had to cut it. And I made something like 30 or 40 different versions of this protein that could be cut in different spots before I finally got one that worked. And that was probably a year of tinkering on and off to try and find the appropriate cut site to figure out what this protein worked. And, I was doing what's called a Western blot, which is a technique where we basically have to develop a picture with film. And I was in the dark room and I remember jumping up when I looked at the film and could see that it worked. And I hit my head on the red lamp that you have in a dark room, the red light that allows you to see a little bit, and I broke the bulb on my head. So that was pretty good.
Worth it. All right,
definitely.
Just briefly interrupt. Scuzzbot, thank you for the gift sub to Calm Down Bronco. Sloth, thank you for the follow. Brawny, thank you for the follow. Brawny, thank you for the subscription. Rebecca as well, thank you for the follow. Again, if you guys want to get further Ask a Scientist event notifications, just follow us and we'll let you know. Eventually we want to do it once a week, but right now due to COVID we have to do it remotely, so Rob is actually joining me via remote green screen from the comfort of his own home. So thank you guys for the follow. Again, T-Man, thank you as well.
It's only been 13 minutes I'm playing this. I just looked up at the timer in the corner.
Feels like a lifetime, doesn't it?
All right. Very good. I just got
SLK. Right. Yes, this game is intense. This is one of the notoriously hard Nintendo games, and if you don't have Game Genie codes, you die 3 times and then you're done. So I agree. It's a tough game. T-Man Gundy wants to know, what's your favorite video game?
Ones without lasers that I run into. Just kidding. Oh, come on. So I think for me, I separate them into 2 eras. So, I got into video gaming when I was a kid, like a lot of people. And, eventually, somewhere in my late teens, I was in college and I was in the sciences, so I had to study all the time pretty much. And I sort of fell out of the loop, for lack of a better term. I stopped playing, oh, come on. So there was a period where I have sort of you gotta be kidding me, I'm like stuck here. I had to do it the lazy way. Or, the smart way. Where I didn't really play for a while. Like probably 10, 15 years as I was developing my career. So for me, things kind of ended around PlayStation 2. And then I just picked up recently, now that my kids are starting to get a little bit older. So we got a 3DS and a Switch. And I've sort of picked back up there. So for me, as far as new games go, the best thing I've played hands down recently is Breath of the Wild for the Switch. That game is absolutely amazing and beautiful. I've already played through it, twice, which I don't have a lot of free time. So for me to have accomplished that means I devoted pretty much every waking minute I had that I could to this game, which I'm sure my wife and kids are not so happy about. But that's all right.
So speaking of gaming history, we're gonna do a quick survey on the audience. So for those of you in the audience, we now have hit affiliate status, which means we get to use imaginary internet points to bet on things. And so if you look in the bottom left of your chat, we have standard internet units. Our first official bet on this is actually going to be, does Robb Tomko remember the Konami Code for this? Don't answer the question yet. This is for the betting. People can bet yes or no. It's been 20 years since you played this game. Do you remember the code? And so in this case, the code is basically early Nintendo games. You put in left up BA combinations to get like extra lives and things. And there's a notorious code known as the Konami Code that was particularly important for this game. And so we're going to see, after 20 years, does Rob still remember the Konami Code? Make your predictions now. I think we have 2 minutes. All right, sorry to interrupt. So let's go back through the questions. AMG says, you have us all intrigued that we sign in to watch you play a game and listen to more Farm after already having Farm today. Thank you guys for showing up. It's awesome to have you here. It's awesome to casually interact about science because that's what we love doing. So thank you for being here. I know Rob feels the same. Other questions? I do. Switch, we did that. Oh, here's a science intensive one. From your website, your lab had developed a small molecule inhibitor for SARS-CoV-2 enzyme. Can you please talk more about that process, assay and techniques utilized?
Yeah,
but for a general audience.
Absolutely. So the short version is we have identified some that inhibit one of the more important enzymes for SARS-CoV-2. It's something I should say is that, there are lots of... what's up with that one? That one was on a different pattern than the rest of them. We discovered this molecule. It probably will never be a drug, which is fine, especially now that there are vaccines coming out, and it looks like they're probably going to be effective. But trained as a pharmacologist, one of the things that I am always interested in is discovering chemicals that do interesting things biologically. So many of these will hopefully end up one day being drugs. But even if not, they're still sometimes very useful tools for biological or biology researchers like myself. We can use them to poke and prod cells in different ways and see what the outcome is to learn different things. So, I didn't make it. So there's an enzyme that the SARS-CoV-2 virus uses to try and evade the immune system related to this protein disposing pathway that I've traditionally been interested in. The protein is called the papain-like protease. And what it does is it essentially acts like a pair of molecular scissors to cut up and destroy a bunch of proteins inside the cell, and also different proteins that are involved in your immune response, so your protection from the virus. And what this allows the virus to do is basically reprogram the infected cell to make viruses and stop making, whatever the cell happens to normally be doing. So it basically turns the cell into a factory for making new virus. And so this protease removes ubiquitin and called ubiquitin-like proteins, which are kind of like the flags for delivery to this protein recycling center. If I'm getting too complicated at any point, just give me a nudge.
We got a mixture of a little too complicated and okay.
Yeah, there's a whole new level of challenge trying to do this. Oh my God, I'm gonna beat 23 minutes.
Yeah, you just got to do it deathless now.
Yeah, while answering science questions, nonetheless, this is pretty good. So basically, this is a pair of molecular scissors for allowing the virus to reprogram the cell to make viruses and to trick your immune system into ignoring it. So basically what we did was we purified this enzyme after we made it in bacteria, so it's not really a threat to us. And then we can mix it with different chemicals that we have. We have what are called chemical libraries in the lab, which is basically a very large array of chemicals that have different chemical groups, different shapes, different reactivities, and we can basically throw the kitchen sink at this enzyme, this protein, and look for something that jams the scissors.
All right, so just pause for a moment. You beat Contra, congratulations. You should probably do another run. That was sub 20, congratulations. A lot of deaths in there, but I think you can do sub 23.
That's how I roll.
But equally important to treating SARS-CoV, do you know the Konami Code?
I do. It's up, up, down, down, left, right, BA, start.
There it is, ladies and gentlemen. That's 9 lives and 9 continues. 20 years later, that is written in his brain from repeatedly using that as a child.
Yeah, so, basically.
Congratulations. Fun factoid. If you go to my website, my research website, and put in the Konami Code, it gives you a special plus screen. So no.
Yeah, useless additional fact. The Konami Code works in a lot of different Konami games, but sometimes it's backward, sometimes the orders are slightly different, but it's always a combination of that basic pattern. So, in my olden days, I used to remember which ones were for which games. Contra is definitely the conventional Konami Code. I think that might have been the first place it made an appearance.
Oh man, so you missed the Konami Code for Contra then. So we have to go with a no on that. So close. Yes. So close. All right, Skullspot, if you wouldn't mind taking care of our imaginary internet points. Yes, I'm sorry, calm down, Bracco. Your internet points disappearing.
All right.
All right.
What
Control R.
Control R.
Top one.
Yep.
R. Try it again.
Right.
See if we can get the time down and the desk down.
Oh, 20 minutes isn't good enough for you, huh?
I mean, we might as well run it a couple times just for nostalgia purposes.
I'm fine with that.
All right, back to the questions.
I am all about incremental improvements. Actually, I think that was my finest efforts in grad school. The things that I did every day, over and over and over again, I would shave off, seconds here and there, and it added up to a lot of time. Just think about how long I would have been in grad school if I didn't do that.
All right. What's your opinion on the COVID vaccine as a pharmacologist?
Yeah, so I want to start off by saying that vaccines are certainly within the realm of drugs, I guess, but I am definitely not an immunologist. So my comments should be taken with a grain of salt. What I can tell you, and some of this is sort of information I've gotten from my immunologist friends. Oh, not the laser. This is the worst weapon in this whole game.
No, there it is.
It looks very promising. So, the response rates are greater than what people were calculating would be necessary in order to generate herd immunity. And there are some relatively recent studies that have come out looking at related coronaviruses. So the coronaviruses, in addition to causing SARS-CoV, I'm blowing it here, there's also a number of them that cause common colds, for example. So we can study them in the population, we being immunologists, not me, pretty comfortably without really putting anyone in danger. And what people have found is that there's a bit of a cycle for these common cold coronaviruses where people get infected, and it's generally at least a year before they can be infected again. And usually that's only after the virus has mutated a good bit. So what I think that probably means for SARS-CoV-2 is that the vaccine should provide resistance at least for a year, probably a good bit longer than that. And the reason for that is this virus mutates, but it mutates comparatively slow to something like the influenza virus, which is pretty mutagenic. So you're probably all familiar that you have to get a new flu vaccine every year because different strains of the flu arise in part through mutation. So SARS-CoV-2 mutates slower than the flu. So at the very least, we should have to be vaccinated far less often than we do for the flu. As far as the immunology goes, I do think I'm sort of at my limit there, though, for that.
To clarify for the skeptics, will you be getting the COVID vaccine?
Definitely. I mean, why would I not?
Just making it clear, we're on record. This will be on YouTube later. Send it to your friends and family who are skeptical.
I get my flu shot. Vaccines are really so simple and so ingenious. Basically, all you're doing is putting a little flag into the body that says, watch out for this, and when you see it, try to... and there's been all this bad press because of only a few poorly done studies that have since been retracted and shown to be not true. It's amazing how much damage it's done.
It was one particular paper, right? It was one that was just devastating the community.
There's, and there were a few that fed off of that, for lack of a better term, that tried to build on the findings of that. But, generally speaking, those studies were really poorly done, and there have been so many... the lead author had
a conflict of interest, right? They had financial buy-in in an alternative.
Also true.
Yeah, there was a lot of things going around there.
There was a lot of problems with that for sure.
Yeah.
So, and the effects of that have lingered now for decades, right? And you're starting to see the worst parts of it, I think, only in the past few years here now. There's starting to be measles outbreaks, for example. There have been a couple big ones in California and New York, where measles was maybe 2 or 3 cases a year, or something like that, if I recall correctly, even 10, 15 years ago. But so few people have gotten vaccinated that it's starting to be able to spread through the community again. And I think a lot of the problem is that people have forgotten how terrible some of these diseases that we have vaccinations for really are. My grandmother had polio when she was a kid, And she was extremely fortunate. She just had stunted growth in one of her legs, so she had like a shoe that was way thicker on one side than on the other shoe. But I mean, there are people that spent the rest of their lives in iron lungs, where they couldn't breathe for themselves. Their bodies were essentially paralyzed, and that can all be prevented by, for polio, you eat a sugar cube, which is delicious, right? I mean, like, and people are eschewing that nowadays, which is terrifying.
Yeah, that's insane. All right.
Pamela Anderson told them it's bad. Which,
with her virology
Oh, and
degree.
just for the record, some of you probably see that I have 32 lives, and that's because of successful input code.
You did the code
I
before?
I feel like I have to redeem myself here now.
That's awesome. All right. What process do you use to map the structure of proteins' macromolecules in the cell?
Okay, so I'm going to address this sort of two levels. So when you say map the structure, I am the kind of scientist who works with sort of individual molecules. So to me, the structure of a protein or macromolecule is the physical shape of one molecule. What is it? How are the atoms arranged? There's a bunch of different ways that we can do this, and they are a bit based on exactly what we want to learn the structure of. One of the more common ways that people do this is called X-ray crystallography. And the way that this works is you will basically get a whole lot of your thing, whatever it is you want to get the structure of, and you'll get it really pure. So you'll remove all the other contaminants from the cell so that you have essentially only this thing. And then you'll subject it to a whole bunch of different environmental conditions until you get lucky, hopefully, and that molecule will form a crystal, just like a salt crystal or a sugar crystal. Probably all you guys did this goofy experiment when you were kids where you hang a string in a super saturated hot cup of sugar and then you get sugar candy. Hopefully somebody knows what I'm talking about. And it's the same basic idea. So once you have that crystal, you can basically shoot it with an X-ray source. And wherever there is an atom, you will deflect the X-rays away. And based on the pattern that develops from those deflected X-rays, you can back calculate what the shape of the molecule looks like. There's a lot of math, but it's really not all that difficult.
Not anymore. Back in the day, that was worth a Nobel Prize.
Back in the day, it was really
Doing
tough.
myoglobin was worth a Nobel Prize.
Yeah. So actually, I think it was lysozyme at Yale, in one of the buildings near where I postdoced. They actually had one of the original models of, I think it was, as I said, lysozyme, and it was like a proper model, like somebody built it out of pieces of sticks and balls, when you see those, and it's like 8 feet high, and 4 feet on each side. It was really crazy. This was so somebody actually sat there and measured the angle of all these things and put this model together. And I cannot imagine how difficult that must have been at the time. And now with the computer, you can take that same data set and solve the structure in probably 30 minutes.
Yeah, and it solves a lot of it for you. It's so automated at this point.
With molecular replacement and all those things.
Mm-hmm.
Snap.
All right.
So it's far easier than it used to be, and it's even been automated in cases. So people have gotten really good at this, and it's been refined and improved over the years. So in terms of so the caveat to crystallography is that it only really works well when the thing you want to study is very rigid. It's not floppy. It doesn't have any bendy bits. So there have been a whole host of molecules that have been very difficult to study structurally because they have flexible regions. So in the past maybe 5 to 10 years, there's been a second approach that's become really, really popular, and we've used this in collaboration with a good friend of mine to solve some structures. And this is called Cryoelectron Microscopy. And this is basically a similar approach where you're looking at deflection of electrons instead of X-rays. But the idea is the same. The difference is in how you prepare the sample. Oops, I didn't go far enough. So with cryoelectron microscopy, the sample doesn't have to be nearly as pure, which makes it a lot easier, because purifying something out of a mixture of, hundreds of thousands of molecules is not easy or often fun. Oh, there we go. And the other thing is you can often resolve, albeit at lower resolution, less of a clarity to the picture, things that are a little bit floppy. So that has really revolutionized structural biology for people who study big stuff or floppy stuff, me being in the big stuff group. So the way that this works is you basically snap freeze a sample, whatever it is you want to get the structure of, in ice, and you freeze it so fast that the ice doesn't even have a chance to crystallize, and that's called vitreous ice. It's basically ice in which all the different water molecules are still sort of randomly oriented. And what that does is it actually allows you to see the structure of these molecules more or less exactly as they would exist inside the cell or in solution before they would be impacted by the formation of ice crystals. You freeze something, and you guys probably all had like a really old piece of chicken or something in the freezer for a long time, and it comes out and it's all crusty and weird. There's ice all over it. So the same thing happens when you freeze biological molecules. So cryo-EM, because it freezes them so fast, it avoids this process. Anyway, you can that's someone from my pharm class. Dr. Tomko, what happens when oxytocin is given and starts to fill ADH receptors? You should be able to tell me, for starters. But let's see. So you would basically have...
I paused. Let me address these in order. Sorry, there's been people waiting.
I don't stay focused very well, do I?
No worries. I'll keep you on task.
All right.
Excellent. So cryo-EM and X-ray, those are the most common ways to do it.
So basically what you can do is you can take a very, very low resolution of tens of thousands or even hundreds of thousands of individual particles with cryoelectron microscopy. And then you can basically overlay them to increase the contrast a lot. Thanks, Rebecca. So that you can get a really high quality, high resolution picture. And because this averages, tens of thousands or even 100,000 particles, it gets rid of a lot of the noise, and it allows you to see things that are a little bit floppy by integrating, lots and lots and lots of information from many different particles. So that's been really big, and it's allowed us to see a lot of the bigger, more complicated stuff inside the cell, with amazing resolution, really for the first time. So I actually started my lab to work on some questions that came about from these original studies on the proteasome. So, we got this sort of really beautiful picture. I am blown it here. It's really
Rob, I hate to interrupt, but we are falling behind on questions.
Okay, no worries.
Yeah. Are you good on that answer?
No, I think that's about it. So those are the two big ones. There's a few other popular or only applicable to a very small collection of molecules. All right, let's move forward. Sorry.
No worries. I'm going to cut you off because your answers are quite lengthy and detailed, and I appreciate that, but we have people that want knowledge. So we will move on to the next questions, particularly from Rebecca Sal 345. Do you think mRNA technology that was used for vaccine can be applied to drugs in the future to permanently fix certain genetic issues like CF? What is CF?
Yeah, so yes and no. So the basic idea does not translate perfectly. So basically, mRNA is a copy of a blueprint that is used to make a protein, right? So basically, the way the COVID vaccines that have been developed is they put the mRNA, the copy of the blueprint, into the cell, and then your cell makes one of the viral proteins that's not dangerous on its own, and your body learns to attack it. And that's how you develop immunity to COVID. So that works great when you can generate a lasting picture of that molecule that you can then store for the future. The problem with mRNA technology, cystic fibrosis, is that the mRNA and even the protein that it makes are relatively short-lived. So what you would need to do in the case of cystic fibrosis is you would need to replace the original blueprint with a functional copy so that you can continue to make new copies and proteins whenever you need to, rather than at just one point in time. So there are ways to try and do that. Gene therapy is usually the name that people use to describe that process. And in that case, via one means or another, you introduce the proper blueprint. So a normal, healthy blueprint for that particular defective gene into the person's cells. And then all subsequent copies of the mRNA and also the protein are made properly, for lack of a better term. So for people that have genetic diseases, single mutation in a single gene that causes the disease, gene therapy really holds a lot of promise to fix those conditions, but we can't really do it with mRNA the same way we can with the vaccine.
All right, so it looks like you're getting bombarded with very specific questions from your students. So I'm going to keep it a little more lighthearted right now. And this question is for me, and I like to ask every single guest I have. So I've seen over 5,000 or so movies, according to Netflix. And my favorite question for an expert is, what movie or TV show gets your expertise wrong, and which ones get it right?
Okay. So wrong is easy. It's almost every movie.
Oh, harsh. Harsh.
I mean, and don't get me wrong, I certainly appreciate the importance of artistic license when it comes to this kind of stuff. There's one movie, and I forget exactly what it's called, but I think Ice Cube was in it, and they go to Mars. Do you happen to know what the movie is?
Oh, yes, I do know. I don't know the name of it. Someone in chat.
Okay.
Someone knows this. They love this movie.
I know, seriously, somebody must know that. Anyway, there's one scene where they're talking about, some DNA that they isolated from something on Mars, and they have like an atomic structure, which is like the actual atoms of DNA up on the screen. And somebody looks at it and they go, that looks like human DNA, which is like completely impossible to tell. I mean, human DNA is completely indistinguishable at the chemical level from any other organism.
Good point.
I just lost the spread.
They could say Earth DNA.
Even that is not necessarily accurate, right? So here's your useless and also risk fact for the year. All the astronauts that have been to the moon have left their poop there. So there's actually astronaut DNA and also astronaut poop on the moon. So that DNA looks identical to the DNA here on Earth. I mean, it's just a chemical, right?
Is the movie Ghost of Mars? Is that
Yes, that's it. Thank you.
Scuzzbot came up with the answer. Thank you.
Come on.
I think you narrowed it down with Ice Cube. All right, so that gets it wrong.
A very bad
I mean, that's fair. All right,
1841.
so who gets it right? I mean, who does Jurassic Park DNA right-ish?
Jurassic Park was reasonably close. There were some things that were inaccurate, but in terms of the process that they described, it would be theoretically possible. Ice Cube goes to Mars, yeah.
That's how you find that answer. Well done.
Yeah. So the thing that wasn't true about Jurassic Park is they extracted the DNA from, like a mosquito that was trapped in amber. And the chemical half-life of DNA, how long it would actually exist, is only like 5,000 years or something like that.
That's in a rigid media, like in a solid glassy state. It's still like 5,000 years or something.
It's only a couple thousand years. So realistically, after 50,000 years, you'd be unlikely to be able to recover anything useful. But we get like woolly mammoth DNA and stuff because they were around only 10,000 years. So it's kind of degraded. But it's good enough that you can still sort of figure it out. It's like reading somebody's handwriting where the page has smears and stuff on it. You can kind of make out what they were getting at. But it wouldn't work for dinosaurs because they were 65 million years ago.
So does anyone get it right, like experimentally or ambience wise, or
completely right? I think probably not. The truth of the matter is, the details are often not interesting enough to be featured in a movie to the general populace.
Sismani, thank you for the $1 donation. You are paying for beer money for scientists. We appreciate it.
Am I doing this again?
It's up to you if you want to move on to the next one.
Oh no! How was my time for this one?
It was about 19 minutes for the last run.
So I did better. All right, let's do one more.
All right.
And then we'll move on to the next game.
I'm assuming this is
I feel like I'm making progress and I have to keep trying.
So I'm assuming this is an inside joke from Emma. Yo, Dr. Tomko, can you take a barnacle shot right now, please?
Can I take a what?
A barnacle shot. I don't know what that is.
No. So now I have to explain this. When I was in grad school, I drank a lot. We'll just leave it at that. And we had a game we used to play where we would go and we would try to drink everything on a shelf at a bar. And eventually we did this at basically all the bars that we really go to. So one night we decided that we were going to we needed a new game basically. So we decided we were going to make up, this is not a good place to be, there we go. We're going to make up a drink that doesn't actually exist and keep asking people for it until somebody, either asks what it is or makes something and just hands it to us thinking we'll think that's what it is. So the drink we decided on was the Rusty Barnacle, which is the name of the sea captain, for those of you who are wondering. And it doesn't sound like something that you would want to put in your mouth. And it's not. So, what happened is eventually people would start to ask us what's in it. So we had to come up with something that nobody would have.
Rob, sorry to interrupt, but Sloth Wrangler, he just spent internet points. He or she spent internet points to have us drink alcohol. So it's time to drink again.
Okay.
I'll put one at like 10,000 for shots of like tequila, but for now we'll do beer.
Yeah, I don't know if I have tequila in house, but I have other stuff.
We'll figure it out. They don't have that many internet points yet.
I will drink anything short of rubbing alcohol if I have it. Don't drink rubbing alcohol.
So your students could have a coordinated effort to give you alcohol poisoning. Heads up.
Yeah. Remember, I'll see you all on Thursday.
All right. Tomko
anyway,
sorry.
So the drink we decided that a Rusty Barnacle was half a shot of Captain Morgan and a shot of tuna juice mixed together. And so we would go from place to place asking for this, and nobody would ever have it. Big shocker. And we went to this dive bar on the south side of Pittsburgh. I did my graduate work at the School of Medicine in Pittsburgh. And no joke, we were the only people in this bar. We're like, hey, can you make us a Rusty Barnacle? What is it? And we explained, and she's like, oh, yeah. And she goes to her purse and pulls out a can of tuna that she had there for, like, her lunch or whatever. Lunch, I guess, food, whatever to eat while she was on break. And she mixes up this shot. And up until about half a year ago, I had done this shot exactly once. It was enough because it's even worse than it sounds like. So, there we go. So basically, the alcohol curdles all the proteins in the tuna juice immediately, and it just turns into this like semi-solid gunk.
Ugh.
Yeah. Anyway, so I did one recently for the second time in my life as part of a happy hour fundraiser type thing for the physician assistant students that I teach. And it was just as bad as I remember it. And with any luck, I will die before I do a third. Not the best idea I ever had.
All right. So we're almost 1 hour in, about 50 minutes in. For anyone just joining us, welcome to Ask a Scientist Gaming, mediocre gameplay and expert level science. Our guest today, Robb Tomko, a pharmacologist who also plays Contra and is happy to answer questions from chat. So Rob, again, thank you for being here. You know your last name, Tomko.
Thank you for not calling my science mediocre.
I apologize. Every time I see your last name, I want to say Tomako. You remember the Simpsons episode?
That's fine. I get Tomoko all the time. People put a P after the M. I get all kinds of variations, which I'm kind of amazed by because it's 5 letters.
Yeah, no, it's phonetic. It's straightforward, but for some reason, my brain... For those of you that don't know, Simpsons did an episode where they grew tomatoes that had infused tobacco in them. All the animals became addicted and people. It was an amazing case study. Simpsons did it first. So
that didn't go well.
Absolutely amazing.
That was an outstanding episode for sure.
All right.
I'm blowing it here.
All right. What do you think of the Nzt Pill from Limitless? We're back to movies.
What do I think about the
Have you
Pill?
Have you seen Limitless?
I haven't seen the movie, but I've seen some episodes of it. So I think there is nothing exactly like that. It would be great if we could create it, obviously. For those of you who don't know, this is basically a pill that increases the brain power of the taker many, 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 many fold. There are a few drugs that are, I shouldn't call them drugs, they're chemicals, potential drugs, that are just starting to be investigated that seem to increase memory in people, which is sort of the first step, in my opinion, toward a proper NZT. One of the better studied ones so far is called ISRIB, ISRIB, and it's kind of weird. It's actually a drug that inhibits what's called the unfolded protein response, which is a pathway that helps to get rid of these defective proteins that sometimes accumulate in the cell. And they gave it to mice, or maybe rats, I don't remember exactly, and then gave them a memory test, and they did way better. So there may be some hope that one day we'll have a proper NZT.
All right, so on record,
on
YouTube record, make a prediction when we're going to have a pill that makes people smarter.
Say it again?
On YouTube record, when are we going to have a pill that makes people smarter? What year do you guess?
2082.
All right, 2082. Let's hold out till then. I will turn 100.
I'll be 102 by then. So if assuming I'm alive. So if I'm wrong, no one will be around the.
Yeah, no one's gonna hold this against you. Oh, T-Man Gundy has redeemed. Take a drink, Rob. Take another drink.
Okay, done.
You guys are destroying his speed run of Contra with your drinking.
I think the alcohol is also doing that on its own.
Yeah, we appreciate it, though. Okay. Thank you all.
No worries, I'm having fun.
Rebecca, thank you for the $1 again. Beer money for scientists. We really appreciate it. Thank you. All right, so yeah, a lot of students in the audience. I'm going to ask, again, lighthearted questions because I'm not an expert. In fact, they're more of experts than me. I like to do the more lighthearted ones. What's the biggest unanswered question in your field right now? Like, what is the next Nobel Prize for your area?
For my field?
Yep.
So that's depends on how I define my field. So, in terms of protein degradation, I think one of the things that's really sort of poorly understood is exactly what the mechanisms are that allow the cell to distinguish one particular protein from a virtual sea of other proteins to destroy it without touching all of the other ones. So, we know some of the very basic rules, for lack of a better term, and we can look at a protein that's about to be destroyed and we can say, oh yeah, that one will go because it has this flag on it. But one of the things we really don't understand is how the cell is able to recognize specific proteins under specific conditions sometimes and leave all the other ones completely unaffected. The slightly more detailed version of that is, we would call that substrate recognition by ubiquitin ligase. It's still something that's very poorly understood. you know, that would probably be my answer.
That's the big one.
It's like a needle. It's like a needle in a haystack, you know?
And the body does it.
Yeah, and it does it with incredible fidelity and efficiency every day, all day. You know, you spend an incredible amount of energy destroying stuff that you spent an incredible amount of energy making, which is really hard to think about it. But, you know, it's just like anything else. You make something purpose and it does the purpose, and then you don't need it anymore. And you don't want it taking up space or potentially even doing things it's not supposed to do when you don't need it around, right? No, I just walked right into that. Could you imagine what my time would be like if I would stop dying all the time?
Exactly. Now you could just get a speedrun record. I'll put it on our YouTube channel. We'll get you on there.
I feel like this may finally justify me channel.
Fair enough.
Watch Rob get drunk and die. Over and over again.
That's what I'm going to title this YouTube video.
That works for me.
Everyone follow us both on Twitter, on Twitch, and YouTube, and you will see Rob get drunk while playing Contra. All right. Any other questions from chat? We do already. It's called Adderall. That's in relation to the Limitless drug, presumably.
Well, you know, here's the thing about that. It works in the short term. It does not work in the long term, right? So, you know, there have been numerous studies on this. A lot have actually been done with respect to methamphetamine, which is basically, you know, it's close to what Adderall is. People's focus and attention and everything go way up in the short term. But in the long term, it goes down because it starts to damage other parts of the brain, and it draws attention away from these things that they actually want to focus on. So it kind of works, but only if you don't abuse it, I guess.
All right, speaking of abuse, so we have a poll for everyone in chat, and Rob, you're going to answer this question. What is the most dangerous recreational drug? Cannabis, cocaine, alcohol, methamphetamine, or synthetic cannabis? Rob, don't answer yet. We're going to have chat vote on what they think the most dangerous recreational drug is. I guess it really depends on how you define dangerous.
Right, so...
Wait. Nope, not yet. We're still going to conduct the poll.
Okay.
Scuzzbot, can you pop that up?
And I feel like we have to say this nowadays. No Googling.
Yeah, exactly. No Googling. We're not going to cheat here. In the meantime, Rebecca Sal has a question. Favorite color?
Favorite color, black.
Done.
So it doesn't work very well down here, but when I lived in the Northeast, basically my entire wardrobe was jeans and black t-shirts. And then I moved to Tallahassee, Florida, which is substantially warmer than Connecticut. And I basically was sweating to death. So I still have a lot of black t-shirts, but now I'm starting to acquire other colors. Because otherwise I'm roasting out in the sun here in Florida. And I don't know if you can tell, but I'm pretty fair-skinned. So I already got that against me.
All right, we have a joking question as well. What is the best Halloween costume you have ever seen, and why is it Freeman as Towley?
Why is it?
Freeman as Towley. I don't know what that is. I assume this is a student in an inside joke.
Why is it? What is Towley?
Freeman as Towley. Oh, all right, we'll leave that one by the wayside.
Yeah, I'm not sure I know what that means.
All right.
I'm having a little bit of trouble making out what the last couple of there were. Something. Is it like Tally, like South Park?
I'd assume so. That's where I default when I hear Towley.
This is probably my good friend from graduate school, Mike Freeman, then.
He dressed up as Towley. All right.
Definitely.
Well done. All right, before we answer more questions, what is the most dangerous recreational drug? Robb Tomko on the record. Cannabis, cocaine, alcohol, methamphetamine, or synthetic cannabis?
Can I enter Contra?
Contra is pretty dangerous. I'll give you that.
I have died a lot. Like a lot.
So your answer from prior information is methamphetamine. Why is that?
Yeah, so if you look at the number of hospitalizations for drugs and then normalize it to the number of people who report using said drug, you have the highest hospitalization rate for methamphetamine out of any drug. So, it has a very narrow therapeutic window. A therapeutic is a term that doesn't really apply to drugs of abuse per se, but it's basically the dose that causes a desired physiological effect. So in this case, I guess it would be getting you high, to one that causes a toxic effect of some sort. So, methamphetamine is really hard on your body. It damages your heart and a number of other organs by activating what's called the sympathetic nervous system. So it causes a whole bunch of parts of your body to work a lot harder than they normally would. This includes things like your heart, your blood pressure goes way up, and you can damage a whole bunch of other stuff too. So that is the most dangerous in terms of hospitalizations, which I think is probably the fairest way to parse. I just got run over by a coal bucket. What is that? What is that thing? Why are they here?
It's alien-based. It's fine.
Anyway.
All right, so your take-home message is don't do meth. Do which one of the list?
Yes. Oddly enough, synthetic cannabis is not all that far behind methamphetamine. And the reason is that it tends to contain a whole bunch of garbage chemicals that aren't really desired and have a bunch of undesired biological effects. So there's a lot of people going in for like that crap sativa or whatever that they buy through the internet and then get terribly sick. So
the lack of regulation. I mean,
yes,
yeah, that's pretty terrible.
Not so good.
All right, so you endorse smoking weed then?
Certainly over smoking methamphetamine or synthetic cannabis. Yeah.
It's becoming legal in most states. All right, Calm Down Bronco has a serious question about cancer. Do you think there will ever be a catch-all cure for cancer, or do you think it'll be mostly situation condition based?
Cancer is this weird sort of blanket term to actually describe any disease where some tissue is dividing when it's not supposed to, right? That's basically what a tumor is. It's a tissue that's dividing and growing when it's otherwise not supposed to, right? Most of us become adults and we stop growing, at least vertically. Around the midriff is maybe a different story, but your body is programmed for most of your tissues to stop growing. And basically every tissue that develops into a tumor does so in a slightly different manner. There are different proteins or genes that become damaged or deregulated that cause it to form. So it's kind of like a whole bunch of diseases that all have something in common, and then a lot of things that aren't in common. So I have to study for Pharm Quiz. Good for you. I'm proud of you. I have 43 lives, by the way. Go me.
You're doing well.
So, the idea that we will get one thing, all of these different things that are only sort of a little bit related is not going to happen, unfortunately. I'm doing worse than last time, aren't I?
So it's going to be a mix of treatments plus there's some preventative things like the HPV vaccine and stuff like that, right?
Yeah. Obviously not smoking is really effective.
Yeah. Environmental influences. Makes sense. All right. Pistol.
Yeah, but go ahead.
Oh, sorry. Pistol's asking, speaking of meth, how accurate was the show Breaking Bad in terms of making it? I can actually answer this one.
Yeah, I'd say you might be slightly better suited for this than I am.
Yeah, as a chemist who does
I can tell you what
synthesis.
I do in the Walmart bathroom with some Drano and some Sudafed.
You'd be surprised what you can get done. But the answer is Breaking Bad is actually really good in their depiction. So they intentionally left out steps that you need to actually reproduce it like it was a recipe. But they consulted experts. In fact, I was a grad student at University of Southern California, and I know the experts that they consulted in terms of actually making the show. So they did really good job with the synthesis and the details and like the methodology. So that's awesome. Like if you asked me what show got it right, my answer is Breaking Bad, because they did a really good job with it. The only exception being it shouldn't have that blue tint associated with it. I know they did that for dramatic purposes, but if you make it clean, it should be transparent. Yeah, exactly. It takes a small impurity, and you might do that for branding purposes and have no, effects on the body. But yeah, no, they did a really good job.
I'm trying to remember, were they doing like the
So they did the methylamine route. So they did reductive amination to make it. Which don't Google any of these words or phrases.
Yeah, or the FDA.
I don't want you guys ending up on a list.
Somebody will be watching you.
But watch Breaking Bad and enjoy it for its scientific literacy. Things like HF, or eating through a bathtub, or the mercury fulminate, the explosive crystalline powder. Awesome stuff. Breaking Bad did a really good job. And they made
1957. I don't know when you stop it.
Oh, I don't know the actual stop time either.
Okay.
I'd have to look it up. All right. MGM wants to know thoughts on pharmacological use of CRISPR, and when do you think it'll begin to be used to cure diseases?
Yeah, so I think the first CRISPR-based, there's some that are in clinical trials already, I thought. My FBA agent would not be surprised with everything I Google for school. Yeah, really.
All right. We should pause briefly. Maybe it's time to step to the next game.
Yeah, definitely.
What do you think? Super C?
Is that the next one?
Yep. That is
all right, what happens when I double click this? Oh, it starts on top of the other one. Hold on. All right.
Oh, man, there's Rob's green screen.
Oh, okay. Do I have to go back to Discord and...
Yeah, you'll have to share it again. Here, we'll go with the cut screen. Does anyone in chat know what the molecule to the right of my head is? Oh, never mind. He's back.
All right. Oh, this is...
Super C.
Yeah, this is Nintendo.
Yep.
I might have to reprogram the controller real quick. Hold on one second. Oh, no, wait a minute. There we go. I don't know what I did.
All right, Super C. So this is the 1992 year sequel to the game we just played.
Is it followed by Super Duper C?
Might as well be. All right, so we're on to the next game. All right, CRISPR to cure diseases. Sorry for interrupting.
Yeah, so, so basically, I think there might even be one that can be used to cure a certain blood disease that's already approved. I would have to double check that. So here's the deal. CRISPR is, I had to move this arrow out of the way. There we go. Is certainly a revolutionary advancement. Hold on. Until I can't talk and shoot people at the same time. But there are still a number of very serious limits, the biggest of which is it will often induce undesired mutations at other sites. So, as I'm sure you can imagine, this is a huge hurdle to getting a drug into clinic, right? Because, you could cause cancer for somebody that way, and that's not good. So, there are a few cases where that risk can be mitigated. The best is in blood cancers, blood diseases. And the reason for that is you can take blood cells out of somebody's body, you can CRISPR them in a test tube, basically, and then you can check to make sure you haven't made any of these undesired mutations elsewhere. I just got shot big time. And if everything looks good, you can put those blood cells back into somebody's bloodstream, right? Just with a simple injection. So that is very easy. What's going to be harder is for diseases where it arises tissue that you can't just kind of remove and repopulate, right? So like if you have liver fibrosis, as an example, let's say, we can take out some of your liver, but we can't really take out all, can't replace all the liver cells you have entirely. So in order for CRISPR to be useful, look at this thing. In that case, you would basically need to get it into every cell in the liver and then have absolutely zero mutations occur in any of those cells, which is practically impossible at the moment. I'm hopeful that the time will come where we'll be able to do that, but we are not there yet. Where are these? Like, why don't they? Why don't these guys have weapons? They're just like jumping back of a helicopter. Does anyone else find that strange? Okay.
There are no rules. This is Nintendo.
No.
All right. Other questions for Dr. Robb Tomko.
Oh no, what's happening? Oh, this is interesting.
Oh
so,
yeah,
again,
the
I
overhead
have not
view.
played this one before, I believe. So
Yeah, they changed up
for
their
a treat.
gaming strategies here.
Get away. Ooh, F.
All right, another question. Has there been a breakthrough discovery in your field in the last few years? Something that changes everything or changed the way you do business?
In the last, what was it, 20 years?
Few years, whatever few means
years.
to you.
Yeah, so for my field, the game changer, this cryoelectron microscopy that I talked a little bit about before.
The recent Nobel Prize.
Yeah, it actually just won the Nobel Prize. And, again, this thing that I study is really, it's like a big molecular machine. And it's very floppy in molecular terms. So it was absolutely not receptive to X-ray crystallography to figure out what it looks like. So you could imagine what it would be like to try and figure out how a car works without being able to see it, right? It'd be like practically impossible. So, we were really kind of working in the dark the same way, trying to understand how this thing that I study, the proteasome, works. And being able to see it, more or less crystal clear for the first time was an absolute game changer. So basically everything we've done in the past 5-6 years since I've had an independent faculty position here has been a direct derivative of these structures, knowing what this thing looks like in the first time. And for many other labs that work in protein degradation, that's the same. Made it. Oh, did you guys see that? I made it right through those bullets.
Nice, well done.
I'm excited. I don't know if anyone else is excited, but I'm excited.
So FSU has a really nice cryo-EM, right? It's like
Yes.
one of the best or top 10 or something in the country.
I believe it. I don't know how nowadays but when I arrived here, they were one of, I think, 7 in the country. So it was a huge deal.
Yeah.
And they still have, they've been upgrading constantly. So they have all the bells and whistles as far as the advancements and technological doodads and all that kind of doodad. That's like an old people word.
Rob, I hate to break this to you, but you are an old person now.
I know. I think you're 40. It's terrible.
You're playing 8-bit Nintendo while playing on Twitch. You are the old person.
I know. There are children everywhere looking at me like old, ugly.
Welcome, children, to Ask a Scientist Gaming with the old people.
I would like to tell you
gaming.
about how I walked barefoot in the snow uphill both ways when I was your age. Also, get off my lawn. Yeah.
Exactly.
But that's okay. Yeah, so that's the biggie for us. It's like being able to look under the first time to figure out how it works. And you're like, oh my god, look at all this stuff in here. So it's been a lot of fun. All made possible by Cryo-EM. These things in the ground are making me angry.
All right, Rob. So one of the questions you sent me is you want to poll the audience as to what is the most expensive drug. When you say that, do you mean in terms of cost or development cost? Because we're going to do a poll quick and survey that. What does most expensive mean?
I actually had to look this up.
Yeah.
Well, there's new drugs all the time, and drugs go off patent and they get cheap, and so as of this morning, at least, this was true.
Well, yeah.
I don't think it changes.
So we'll put up a poll for the people in chat. So when you say most expensive, is that development cost or is that?
Are you asking me?
Yep.
That's in terms of so if you were to go to your pharmacy and pick up your prescription, what would you pay out of pocket? If you had no insurance.
All right. So the poll is up. What does the audience guess? What is the most expensive drug? How much does it cost if you didn't have insurance and you actually had to pay for it at cost? What is that going to be? Vote now, please. Keep in mind, any of our international guests that don't have concerns about healthcare, pretend you're an American and answer this question accordingly.
What are you talking about?
Exactly. What is one unit of treatment in America?
About a failure of American healthcare.
Exactly. This boss is something else.
Also, PA students, it's none of the ones you past couple of weeks, just to be clear. Don't waste your time.
We're going to give them a hint. Is it a modern one?
Yes. It's past couple of years, I think. What is the deal? Am I not shooting? Oh, I'm not shooting in the right place. Okay. Why aren't you dying?
Oh, see, you got to hear that sound. That's the you're hitting it sound. Yep.
Ugh. Also, who built this thing? Like,
I'm assuming the aliens, but I don't know for sure.
Yeah, I don't actually even know who I'm fighting in this game. I'm just willing to shoot anyone without no...
All right, last chance on the poll. What is your guys' guess is the most expensive drug if you were to try to just buy it and use it right now without insurance?
Oh, yeah, it was good. We could probably do away with the timer.
All right, the guess from our audience is $212,500. What's the real answer?
You asking me?
Yes. Well, if you want to answer it, I have the answer here. If you want me to give it.
Yeah. So it's a gene therapy for spinal muscular atrophy. It's over $2 million for a treatment regimen.
So 2.125 million is the correct answer for a drug treatment. That is crazy. So, how is that even a real number? Like, no one can afford that.
I don't know, but I can tell you this much. Go in and see has change for $1 billion. The answer is no.
That is intense.
What are these things?
All right.
Yeah, so, I guess on a positive note, it's a gene therapy. So once you get it, you're theoretically cured. So you only have to pay that once.
Then you're fine. Oh, that is crazy. It's not like
picking up your anxiety.
I mean, so that's so there's a scaling cost associated with things, right? So this is presumably some kind of rare disease where only some people get it.
Yeah, so this is a relatively rare disease. And if I recall correctly, this actually only fixes a particular form of spinal muscular atrophy that arises in a certain way.
I see.
So, it's highly specific. There's probably like 100 people tops. Oh, look, Netflix has a documentary on this. Very sad. That sounds about right for America.
MGM, thank you. I'll have to watch that one as well. Osmosis Jones in a Netflix documentary about
do you know what it's called?
Million.
Now I want to watch.
Yeah.
What is it with bubbles in these Contra games?
It's alien-based.
Has anyone ever been hurt by a bubble?
All right, $2.1 million if you happen to have that disease. That's crazy. It's called unnatural selection.
Oh, you know what? I actually watched this. How about that? This is the one, it was actually talking about people who were doing CRISPR like in their sheds behind their house.
All right. So you have a recommend from Chat and Rob to watch that movie, Unnatural Selection.
Yeah, it actually was a good documentary. I enjoyed it at least.
CRISPR in a shed. Somebody clip that.
I'm not kidding. I mean, there are people, they call themselves biohackers, and they think they can go in and modify their own genomes and stuff. And, as someone who uses these approaches in a laboratory, I'm listening to the way that they're doing it. I'm going, oh my God, you idiot. This would never work.
I mean, it'll work, just not the way they want it to.
No, it won't work most of the time. But if it does, it's definitely not going to be good.
Yeah, no. I mean, if you want cancer, I can make that happen.
Yeah, seriously. It's
There's many things
easy
you can eat.
Get.
Yeah.
Actually, it's not even that easy to get cancer. I mean, in the grand scheme of things. People don't realize this, but your body has an incredible system set up to avoid developing cancer. And the average person gets hundreds of tumors in their lifetime that are eradicated by the immune system without them ever knowing. Which is kind of terrifying, isn't it?
Bronco asks a follow-up question that actually preempted our question. Aside from materials and equipment used to make drugs, what causes a drug to be unreasonably expensive? Is it just corporate greed, or are there other things that go into it?
It's a combination of things. So, at the end of the day, something that I think a lot of people get is that the people who make the drugs are businesses first, right? And in my opinion, the fact that they decide to make a product that has a noble use is honestly good. So here's the thing. The average cost to get a drug to market, to discover a chemical that does something useful, show that it's safe in people, and that it actually accomplishes whatever the biological goal is in people, cure a disease, treat a symptom, whatever, it depends on who you ask, but it's on the order of like $1.5 to $4 billion. Okay, that is an incredible amount of money, right? I mean, most of us will never see even $1 million. Well, hopefully we will, but a lot of us will probably never see $1 million in our time. So multiply that by 1,000, and that's what it takes for somebody to actually develop a drug that's safe and effective and gets approved by the FDA. So here's the thing. That drug gets patented, right? And after a couple years, anybody can make it and sell it. So the truth is, the way the system's set up, a company spends an incredible amount of money to identify and develop a drug that works for something. And then they only have a few years to make that $1 billion+ back and make some profit so that they actually can be functional as a business, right? So that is part of the reason that drugs are very expensive, is that many of them require an enormous investment in research and development to make it to market in the first place. The second problem is basically the U.S. health insurance system, which allows insurance companies to basically, and drug companies and everybody else to basically set their own price however they see fit. So, if you buy certain things from a hospital out of pocket, it's, let's say, $125. But when they charge your insurance company for the same thing, it's $3,000. And nobody says, why are you doing this? That's not fair or reasonable. So that's the other part. And depending on the drug, you're getting it in a hospital, so you're getting that additional inflationary charge. Okay, what do I do now? Do I keep walking? Oh, no, explosion. Super duper. So those are the big parts. And if you look at things that have been around for cheap, you can buy aspirin super cheap. You can buy omeprazole, Pepsid AC, which was once a prescription drug, over the counter for $20 or whatever at Costco now, right? So, once there is competition for that market, the price goes down. But the system is set up so that these companies have to put a huge amount of money in to make a product that can earn money, and then they have a very small window to actually make that money. So that's a big problem, in my opinion.
And so Emma has directly related follow-up. Shouldn't costs go down as the patent expires and then there's competition? Why doesn't competition drive costs down if there's a bunch of competing off-brands?
Right. So in many cases it does. And, I just gave a good... All of you have gotten things like, amoxicillin or whatever for $4 at Walmart or Publix or wherever, the grocery store, wherever you pick up your prescriptions at. Those things used to be super expensive when they were new, and the company needed to, I have the hiccups from drinking, to recap their money from the investment that they made to identify it in the first place. Where on Earth are there purple boulders?
We don't know if you're on Earth.
No logic in Contraland. And now I'm on top of a train, because why wouldn't there be a train at the top of this mountain thingy I just climbed? Yeah, that's a good question. And to some extent, the other thing to keep in mind is some of these chemicals are unbelievably expensive, difficult, etc. to make in the first place. Just to physically make them. The starting materials are super expensive, or they require an incredible amount of effort and quality control and things, and that also drives up the price. So there are, of course, some things that are generic and are still expensive, but usually there's a reasonable reason for that.
So a counterexample and a question here. Competition doesn't seem to apply to insulin. Why is that?
No, and that's an outstanding example. Insulin is actually super cheap to make. There are a few small complications, but you know, people grow it in yeast. They basically engineered yeast, baker's yeast or you know, brewer's yeast to make insulin. So in principle, it could be very cheap because you can grow yeast in your kitchen, you know, for pennies.
All right, just to make a note now, we can make yeast and make insulin in our kitchen and CRISPR in our shed.
Yes.
We have a home development kit in the works.
I feel deliciously...
That's awesome.
I don't know what the term is. Watch out. What is going on here? Anyway, so that is a good example. And the major problem is, is that there's only one or two companies that have bothered to make insulin. So they can basically set their own price. This is a weird case where, for some reason, no one is making, for lack of a better term, I think, a number of generic insulins. So, like, most of it, I think, still comes from Pfizer, for some reason, if I recall correctly. Don't quote me on this. So there hasn't been much competition. But you're right. I mean, people are literally dying to get their insulin, and I could probably make it in my lab for, you know, pennies. Obviously, I'm not FDA approved to do that, but the truth of the matter is somebody could make it super cheap, and they're not. And that is a huge problem.
At least not in the U.S.
Not in the U.S., if that's fair. You know, so this is where, in my opinion, really should step in to try and stimulate some competition there between companies to make it, right? Either by subsidizing startup costs or telling them that they'll offset, you know, they'll give them a bonus for every, you know, 50 grams of insulin they make or whatever. But there should be somebody trying to make sure that that need is met, and there's not. So, you know, we all have our differences in terms of where we think the government should and shouldn't act, but, you know, I feel like this is something that's so simple, and there's so many people. I mean, it could be done very easily. What am I supposed to be shooting here? Anybody know?
Oh, you might just go. Walking. Yeah, there you go.
When in doubt, just walk.
Yeah, Danning just mentioned the EpiPen issue as well. Somebody had control of that market.
Another example. There's only one company really that makes small. You know, some drugs you can make them and you can sell them at a substantial inflation, and they're still reasonably priced and whatnot. But, you know, I guess that's not the case for some drugs, and companies realize that they're the only ones that are interested in making it, and they charge as much as they feel like, and it's...
I just want to take a moment to appreciate... The way capitalism works, unfortunately. I want to take a moment to appreciate how amazing that boss was.
Yeah.
that I've never played this game before. That was amazing. All right, flashing back to a previous question.
And we're not done.
Oh, there it is.
Giant snake coming out of this thing.
The upgrade.
Why not?
Level 2. All right, TCS Sista. Can you explain prions and why have they been impossible to treat?
Can I explain prions and
why they're impossible to treat?
Right. Okay. What? Was I there? I didn't even realize I was alive again.
No worries. Infinite lives.
So a prion is... these things fall. A prion is basically a protein that can replicate itself. And that's sort of a misnomer. It doesn't have babies, for lack of a better term, you know. But what it can do is it can take a pre-existing protein that is normal, and it can convert it to the prion form. So it makes it change its shape and become contagious. So examples of this are like Creutzfeldt-Jakob disease, chronic wasting disease in a variety of different animal species. This is actually a huge problem in, how about this? 3 dimensions. Or 2 dimensions, I guess. Anyway, this is a huge problem in Pennsylvania. I grew up in Philadelphia right now. It's a huge problem in Philadelphia. A lot of the deer have chronic wasting disease, so people are hunting them and then eating them, and there's worries that in a couple of years they're going to start to develop prion disease. So the short version is when a prion replicates, it causes other copies of a protein to take on this unusual prion shape. The prion shape is usually very, very stable, so it's very hard to get it to switch back to the original form. So I'm sure that someone somewhere is looking at chemicals that could be drugs to do this, but at the moment, no one, to my knowledge, has been successful. It's a very good question. So it's almost like a virus, but specifically for proteins or for a particular protein. We don't know how to undo it yet. What are these purple things I'm shooting at? Oh! Look at this. That was probably the spread. I feel like if this was real life, I probably wouldn't want to be this far down into this collection of eggs by myself. But where's the fun in that? Oh, give me... I hate this gun.
All right, so Bronco has another question. Why do we use drugs that have similar structures to proteins / macromolecules to bind to cells rather than just using actual molecules itself?
That's a good question. So I'm going to give a general answer, but in some cases there is a... For the sake of this, I'm just going to give a general answer. The average chemical, the average small molecule chemical drug has probably 20 atoms, let's say 20, 25 atoms in it. And if you think about all the possible shapes that you could make out of those 25 atoms, it's really quite limited, right? It's probably a lot. It's probably a couple hundred, but it's not that many in the grand scheme of things. So the average protein has probably on the order of 30,000 or 50,000 atoms, okay? So what that means is they are far, far bigger, and they can assume a much greater collection of shapes. So most drugs work like a key in a lock, okay? They very simply plug an opening or a functional site or something on a protein or a macromolecule. So you could think about it, if you have some random lock, let's say you're trying to break into somebody's car, because why not? If you have no idea, you don't have the key and you have to try and break in, you have to take a bunch of keys to get in. You have a much better chance of getting in if you have 50,000 keys, 50,000 different shapes that you can test, versus if you have, a couple hundred shapes that you can test, a couple hundred keys. So put another way, having a much larger molecule allows us to generate far more shapes, far more keys that we can use to plug different locks. So there are many things that have a shape where it's just impossible for one reason or another to dock a conventional chemical drug into. But it's possible for a protein, because usually it's much bigger, to be able to adopt that appropriate shape to stick to it.
You held out there.
Like obvious ripoffs of the xenomorphs from the Alien franchise.
When did Aliens come out?
Just throwing that out there.
Alien movie.
Somewhere, what's his name? Yep. Ridley Scott is... well, he's not dead, but if he was, he'd be rolling over in his grave.
So Alien was 1979, Aliens was 1986, this came out in 1990. So you are right, yeah. It's a ripoff.
How do I get across this?
I'm assuming it's just...
anyone... Oh my god, that's a long jump.
Yeah, it's just timing that hitbox.
Heat laser. Oh my god, I'm the worst.
I like it. All right, any more questions from chat? Otherwise, I'll interject some of my own. Scuzzbot asked his favorite question. What is your favorite formula / equation?
What is my favorite formula /
equation?
Equation. I'm partial to Beer's Law. I use it most days.
Beer's Law is an awesome equation. What is
Right?
Beer's Law and why do you use it?
Right. So Beer's Law basically says most chemicals, not most, but many chemicals, light. And the amount of light that is absorbed in a solution. So if you dissolve a chemical in water, a solvent, some sort of solvent, alcohol or whatever, and then measure how much light is absorbed when you shoot a beam of light through it. The amount of light absorbed is directly proportional to the number of molecules present in that solution. So put another way, what it allows us to do is measure the absorbance, how much light is absorbed. Oh my goodness, that was interesting. For a solution of a molecule. And if we know that molecule in something called the Extinction Coefficient, which we can figure out if we don't otherwise know it, we can calculate exactly what the... So we use this pretty much every day to calculate the concentration of different proteins and biomolecules that we work with in the lab. We can stick them in an instrument called a spectrophotometer. We can measure how much light they absorb, and then we can know how much of whatever molecule it is that we actually have. So we cannot live without this, basically.
What's awesome about that is I work in solar cells and my answer is pretty much the same. Like we literally use this every single day because you have to absorb light. I mean, you have to know how much is absorbed.
We cannot live without it.
Yeah, no, awesome answer. I like it. When I did this, I didn't have a good answer, but Beer's Law is a pretty straightforward. That's the way it should be. In fact, even when we do femtosecond ultra-fast measurements, we still do Beer's Law, just derivatives of it. So that's really fun.
Yep. I believe it.
All right. Other questions from chat? If not, so we take biology in high school. We learn things like, mitochondria is the powerhouse of the cell, and, catchphrases like that, which is true. Is there there's a lot of amazing machinery in the human body or in biological things in general. Is there any example of machinery that just blows your mind, either human or otherwise based, that people should know about just because it's insanely interesting?
Yeah, I mean, I think there's an enormous number this looks like this. Oh, not anymore. I was going to say the top part of that looks like the space jockey from Alien.
Just straight up ripping it off.
Yeah, I'm going to have to write a strongly worded letter to Konami after this.
A cease and desist with a time machine.
Yeah, we're on to you, buddy. We don't really discuss them this way a lot, but your body genes, for lack of a better term, inside it that do different things. We can think of them both at the level of organs, but even within your cells, individual molecules that function really like machines. And some of them, the things they do are unbelievably complex. So as I said, I'm biased again, but the thing that I study, the proteasome, I think is an incredible example of this. This is something that can take virtually any protein that exists in the cell, and they differ in so many ways: size, shape, chemical makeup, electrostatic charge, whether they're slightly positive, slightly negative. I mean, so many different ways. And this thing can recognize them only under certain circumstances. It can take them, they're usually folded into some important shape. It can unfold them and then chop them up into little bits that you can use to make new proteins. And all these processes have to happen in a very carefully orchestrated, stepwise fashion, like an assembly line. And it happens without screwing up. It's amazing. It's amazing how efficient it is. You are a scientist gaming FA. Thank you. I'm flattered. Things like the DNA replication machinery. You have to make an genome every time a cell in your body divides, and there's trillions of cells in your body, right? Did I just beat this?
You did.
I didn't even notice.
Crest Uncle, thank you for the follow.
For reals.
All right, so you want to try this one once more, or do you want to switch to the next one?
let's switch to the next one. I'm feeling saucy.
All right, let's do it.
Contra Force?
Contra Force, the 1992 release, the third of the NES series.
All right. So, I don't know if it's best to do it this way.
You can just drag and drop it or open it.
Okay. Look how organized I am. Are you guys proud of me?
I think you've impressed your students with your game playing / science discussing prowess. This is a non-trivial skill set. Well done, sir.
Thank you. If only I could get paid to play video games. I need a Twitch page.
Yeah, exactly.
Is that working?
Yep. Contra Force.
All right.
The third of the series.
Oh god, I have to pick a person?
So this is one of the last games that, or not one of the last, but this came out basically when the Super Nintendo was coming out. So this was the tail end of NES games.
Gotcha. So should we take a poll to see which character?
Oh, should we do it? Colin, can you set up a poll? Burns, Smith, Iron, or Beans? If I was the chatting audience, I would pick Beans.
Beans. I'm kind of, I don't know, Burns, he has a grenade. Beans has like kind of a weird weapon array, right? He's got what appears to be a clip mine, maybe a P7, and then I'm not even sure what that thing on the right is. It could be his lunch. You know what? Let's find out. Let's pick Beans and see what happens.
Never mind. The audience does not get an input on this. All right. Sorry. Oh, nope. It's coming up. We're going to do the poll. We'll see what people say. You might have to restart depending on the decision.
Okay, that's fine.
All right. The poll is up. Which character should he play as? Burns, Smith, Iron, or Beans?
I'm going to make a probably wrong prediction that it's going to be 100% Beans.
We're going to find out. We'll give people another minute to decide.
From now on, I'm only playing characters named after food.
So have you ever played this one before, or is this a first?
I have not. This is, oh my goodness.
Yeah, this is a blind playthrough of Contra Force.
Attacked by a forklift. The aliens have mastered using industrial equipment.
Respect. If they can do space travel, they can run industrial equipment. All right.
I don't know if you ever tried to drive a forklift. It's not that easy.
All right, we're going to close it with 50% of the votes. Beans wins.
Yes.
you chose correctly.
Just keep going then?
With Beans. Yep, just keep going. No need to reset. But we do have a question.
Musical Contra character.
Exactly. I've heard lobsters are essentially immortal due to the telomeres being regenerated by telomerase. Are there other major factors that go into this amazing aging phenomena?
Yeah, so, all right, apparently I'm going down there. So, I am going to preface this by saying I am not a lobsterologist. Last time I checked, there's probably a more precise term for whatever that would be. So I don't know a whole lot about lobsters, but I do know a good bit about telomeres. So telomeres are basically the very tippy ends of your chromosomes. And the thing with telomeres is they cannot be replicated completely every time your cells divide. There's fans. What's going on? Oh, man. And so what this means is basically your chromosomes get a teeny bit shorter.
Let's pause to appreciate his floating torso was just floating above the fans.
I usually float when I'm dead. It's awesome.
This is realistic physics here on Contra Impact.
I don't know. I feel like the fan's not spinning fast enough for me to be going this. Just saying.
This is amazing.
My bullets? It's something.
I mean, you're aiming while spinning. Somebody's spotting really well. This is amazing.
I stepped on a mine. Anyway, so basically what this means is every time I'm dead, you lose a little bit of your DNA, which is kind of scary, right? So the truth is the telomeres themselves, the sequence of them is not really important. They're just there to protect the important parts of your DNA. So when your telomere, when your chromosomes shorten every time you divide, you're losing something that's not really important. So most species cannot elongate their telomeres when they divide. So they basically, their chromosomes just get shorter and shorter. And when they finally start to chew into the important bits, the cell undergoes what's called senescence, which is it decides that it's never going to divide. Oh, I'm over here. It's never going to divide again. And this is one of the ways that we protect ourselves against cancer, actually. That way we're not replicating an incomplete or a damaged copy of our genome. This is weird. It's starting me to the right. So we don't have to, it decreases our risk of cancer. So some organisms are able to elongate or extend these telomeres when they get short. It sounds like lobsters are probably one of them. So what this means is, in principle, the cells within a lobster could divide forever because they can replace those telomeres and prevent themselves from losing important genetic information. Now, the problem is there are other risks independent of that. So, for example, your body does a very good job of this, but you do occasionally make mistakes when you're making a copy of your genome for cell division. So over time, you start to accumulate these mistakes or mutations, and in many cases, this is what can lead to cancer when you happen to get just the wrong mutations and just the wrong parts on your chromosomes. So even if you are able to divide forever, you run the risk of eventually becoming prone to cancer because you're going to accumulate more and more mutations over time. So what I would be interested to see, what do I do here? Oh, here we go. With respect to these lobsters is whether, you know, they have some particularly effective means to protect their genome or to catch these errors and fix them. There's also, I forget what it's called, there's a certain jellyfish that appears to be able to live literally forever. It's a Japanese jellyfish. This guy has studied it. There's this one guy that has been keeping jellyfish like an unbelievable period of time in jellyfish terms. I guess they usually don't live very. This one's been alive for like 70 years or something crazy like that. Like, what kind of gun shoots bullets? They'd only go like a third of the way across the screen here. Also, is this Crocodile Dundee? Like, no one listening knows who that is.
Doesn't matter. I know who that is.
Thank you. You make me feel less old.
Did you know they made a 4th movie recently of Crocodile Dundee?
Are you serious?
Yeah,
is he alive?
Yeah, he's alive. And the movie's about Paul Hogan, like, dealing with his characterhood of Dundee. Yes, classic gameplay. Or classic movie.
Super duper. Anyway, so this is a knife. Nice, somebody knows. I want to see lots of him and then suddenly never see him. Right? If you're the boss's wife, Burns. I'm not Burns, I'm Beans. This guy doesn't even know who I am. Do you know who I am?
It's quality dialogue. All right. More questions. So we talked about anti-vaxxing. One of the questions I literally ask pretty much every guest is, what is the flat earth / anti-vax equivalent of your discipline? Is there something other than anti-vax that, Is there something other than what? Like anti-vax that your field is commonly dealing with, like people that are skeptical of, natural drugs, homeopathy.
Yeah. I mean, I think the biggest problem that... Oh, I fell in the water. How about that? I think the biggest problem that we face is people not understanding why they are prescribed medicines and why they need to take them.
That's fair.
Like, this is especially common in people. They tend to be a bit more stubborn and less willing to listen to modern medicine than younger people. And, they call this patient compliance, which basically means you tell them what to do to fix a problem or keep themselves healthy, but then they have to actually do it, right? So, a great example of this is, I think, people who develop metabolic syndrome and start to develop type 2 diabetes, especially in the early days, you can actually reverse that in many cases. So no matter how much damage you have done, if you start to make the appropriate lifestyle changes, am I going to get chopped up by these blades? No, I can walk on this. Awesome. You can reverse course, and you can basically go back to normal. And all you really have to do this are the two things that you guess. You have to eat right and exercise right, so that your body learns how to properly manage incoming nutrients. And you can basically reverse the onset of type. Can I go in here? Yes. Okay. You can basically reverse the onset of type 2 diabetes, but nobody does it.
You just walked into that building and got run over. Awesome.
Because,
Anyway, sorry.
Everyone drives, a quarter-ton truck inside a building.
Yeah, the two-dimensional warehouse. I get it.
Yeah. Anyway, so at the end of the day, how about this? At the end of the day, basically, no one actually listens to advice, for example. There's very few people who actually listen to the advice and do what they're supposed to do, even though they're told exactly what they need to do. So, I think that's probably the biggest issue.
So I brought up homeopathy. Are you familiar with this medicine strategy? It was big in Europe. It didn't hit in the U.S. as much as it did there. Are you familiar with this?
I don't know a whole lot about it.
The foundational philosophy is that you take some kind of drug or toxin or something and dilute it down, 100,000 fold. All of a sudden it's more effective than it is as a concentrated agent.
What?
And this was approved by the British medical system as some kind of legitimate. Yeah, no, it's absolutely the founding philosophy behind it is
not how chemistry works.
Yeah, exactly. So the philosophy behind it is that water somehow has memory of this drug, and that memory somehow translates to treating it within your body. Yeah, no, it's absolutely insane.
Yeah, no. I'd say, yeah. No, so thankfully I haven't.
No, that's amazing. I'm glad it didn't take off in the U.S., but in Britain, it was approved healthcare for a period of time. I don't know if it is anymore.
I feel like maybe it's only a matter of
somebody's gonna. Oh man, this is the stream that brings it to America. We're at fault.
I know I'm gonna be inadvertently responsible for something terrible here. To all of those you listening, that doesn't work. I think any of you who have made it a concerted attempt to get drunk know that if you dilute your beer 1,000-fold, you do not get drunk. Okay? Medicine is exactly the same thing. It works on all the same principles. You have to appeal to something people will understand, right?
Yeah. No, there's an amazing Mitchell and Webb clip that tackles that exact issue.
Really?
Yeah.
What is this thing? Oh, he looks like
Mitchell and Webb Homeopathy. Colin, if you wouldn't mind putting that link in there, you guys should watch it. It's amazing. Because after a hard day of administering homeopathic medicine for minor diseases, they go to the bar and have a drink of homeopathic beer.
Seriously.
Yeah, my favorite quote is Tim Minchin. He's an Australian comedian. Water has memory, and whilst this memory of long-lost drop of onion juice seems infinite, it somehow forgets all the poo it's had in it. Which is true. Water has had blood and poop in it in addition to whatever drugs people think they're taking. All right, Bronco wants to know, are there any Eastern alternative medicines that you think actually help outside of being placebo?
Yeah, I mean, so, I think aspirin is actually a great example of this. So aspirin was originally administered by taking the bark of a willow tree and boiling it and then drinking the resultant tea. What is the deal with this guy? Can I go up on the side of him and just shoot him from the side? And people didn't know how it worked for a very, very long time. Not until I think the mid-1800s, but they knew it worked. And the reason is the bark of a willow tree is rich in salicylic, which is almost aspirin, for lack of a better term. And, there's a whole bunch of Chinese herbs, and there have been a variety of different natural products, they're commonly called, that have been identified in different plants that people have been using, as teas or whatever for hundreds of years sometimes. And there's a huge body of evidence showing that they work, and people just don't entirely understand why. And most of the time, excuse me, if somebody goes and looks, they find that this is a drug and it does something that's, biologically productive or biologically useful. So there's a whole host of these sort of anecdotal, herbal, whatever remedies that people have used over the years, and in some places still use, that are clearly effective and just haven't been studied to the extent where they can isolate the particular chemical or chemicals that does the desired process and put it into a pill rather than, boiling some weird plant or whatever. So, some of them definitely do. There are, unfortunately, I think, just as many, if not more, examples of this where people are working more on superstition or placebo. That looks like it's going to hurt me if I poke it. This isn't good. Oh, I'm going down.
Oh, you got to take it again. Oh, brutal. So I'm reminded. Oh, go ahead.
I was going to say, most elevators, but that's fine.
So I'm reminded of another Tim Minchin quote. Do you know what they call alternative medicine that's been proven to work? Medicine.
Yeah, I knew it. I don't know who this guy is, but I'm going to have to look into it.
Oh, yeah, I know. He's an Australian comedian. He's very clever, like social commentary, very science-based decision-making. It's pretty amazing.
Oh, come on. Who leaves lines there?
Honestly.
All right. Now, where do I go? Oh, okay.
All right, we are almost at the 2-hour mark of gameplay. When was the last time you played 2 hours of video games straight?
When's the last time I did anything for pleasure for 2 hours straight?
Well, it's been great having you. Anyone just joining us, it's Dr. Robb Tomko, pharmacologist, playing Contra, answering questions from the chat. So if you have any questions...
I should probably go on the record now as saying I'm willing to do this whenever as long as my wife won't divorce me.
Nightly basis. No, it is a lot of fun. It's fun to revisit these games.
Definitely.
Oh, if you had to choose another series to play, which would you choose?
Yeah, I mean, as far as Nintendo goes, you name it, I've probably done it. Where's my elevator? Now I'm in trouble. This is going to be one of those cases where I walk this way and then magically walk back and it's going to be there. Watch.
You have figured out the rules of this Contra Force
Times
game.
were simpler back then.
Yeah, no soft locks here. Needs to eat your quarters. These games are amazing.
I know. It's funny. They take up like 8 kilobytes or something ridiculously small now.
Yeah.
Okay, I still don't know where I'm going though.
The amount of joy these games brought to people.
Oh my goodness. What am I... I'm a little bit confused about what I'm supposed to be doing here.
I don't know. I can look it up if you'd like.
Let's. Oh, good. These guys are back too.
The infinite bad guy nature of the
They've
Nintendo.
come back to life. And down I go.
You might be able to ride that one all the way up.
I like an elevator that goes all...
All right, so we've done many like science-centric questions and pop culture-centric questions. Now we're going to do some Rob-centric questions. So 5-year-old Rob was not like, I'm going to grow up and be a professor that studies pharmacology. What path led you to where you are today?
I was that kid that was always like taking things apart. like always taking things apart. And I was all about Legos and, building and understanding how, so I was always interested on the insides of things. And, I think that I was sort of doomed to be in some sort of, I don't know what the right term, exploratory career path no matter what I did. But I was always interested in like mechanics and things like that. So even now, one of my hobbies is like, building and rebuilding old motorcycles, stuff like that. So I still like to kind of take things apart, see how they work, and then put them back together. So, the seeds were always there, I think. I was, a reasonably smart kid, I guess, when I was little. I'm really kind of stuck here. We're going to give this another crack, and then we might have to get some help from outside.
No worries.
But I was sort of like the first in my family to college and obviously to grad school. And so I didn't have a whole lot of direction, and I really just kind of followed the things that interested me, which was, at the time when I was in high school, it was chemistry. My high school chemistry teacher suggested I major in biochemistry instead of chemistry at the time because he thought it was going to be more lucrative. We can debate that, of course. My chemical engineer friends were making six figures right out of college, but whatever. Anyway, so I got into biochemistry. I sort of got a taste for pharmacology, exploiting chemistry to do cool biological stuff. All right, I am completely stuck.
What is going on here?
Doing.
All right, I'll look up how to beat this portion. Scuzzbot says, you sound like an engineer. That's fair.
In a lot of ways, we kind of are. So even, a lot of the experiments we do, we are engineering features or situations or whatever. This is like, why do these guys keep shooting me? Do I have like a super weapon or something? What happens when I hit? No, nothing. Weak. I didn't know if I had like a bomb or a missile or something. We use principles of engineering to ask biological questions. We engineer proteins that have unusual features so that we can ask something, and we sometimes introduce completely unnatural chemicals or amino acids and stuff into proteins to change their function so we can ask something interesting. There's a lot of engineering, I think, to scientific experimentation. Come on. Oh, it's back. So I agree. I think said this to actually found out I'm getting promoted with tenure. So for those of you who don't know what the life of a professor is like, the first 5 or 6 years, you're basically working feverishly to prove to the university that they should not fire you. So I just found out I'm basically not being fired, which is very exciting. But I kept telling my wife that if I did end up getting fired, I was basically going to become a mechanic. Which would have been the same thing, right? It's just instead of working on biology, I've been working on machines, figuring out how they work, why they're not working properly, and so on. Same skill set, different medium, for lack of a better term.
Nice.
Congratulations,
by the way. So you got your official letter, departmental vote.
Say it again.
Is it a official letter? You got the university?
Yeah, from the president.
Congratulations.
So I actually have it framed, and I'm going to tell you why.
All right.
It's because it's a blank slate, fill in the blank letter from the president of the university. And my name is Robert J. Tomko Jr. Father, of course, is Robert J. Tomko. Mm-hmm. And my letter awarding me promotion and tenure says, Dear Dr. Jr. So I highlighted it and framed it, and it's hanging in my office. I thought that was fantastic.
That's amazing.
I'm not even upset. I actually thought it was pretty... I give up.
Oh, man. That's like the postdoc application letters I get. You probably get the same emails, right?
Dear esteemed professor.
Yeah.
Where it's in a different font and font size.
My all-time favorite one is I got an email that said, Dear Professor, and then instead of my name, it showed like, it was like HTML code to auto-insert a name or something like that. It was like brackets, HTTPS, blah, blah, blah. Like they had a script to do it, but the script failed. I'm like, that's awesome. Way to go. Generic
That's so
email.
lazy. I love it.
Yeah.
I love it. Yeah, so this is basically the equivalent of that. It's like the president doesn't even, he didn't even bother to actually write my last name properly in the letter.
Ouch.
So I'm forcing my lab members to call me Dr. Junior. Some are taking to it, some are not, but I thought it was pretty funny. Okay, so this thing, I had to shoot this, I think, maybe.
Oh, wow. Okay, so you can change characters.
Oh, I did it. Oh, thank God.
There it is. I just found the video.
All hope.
Sorry. Should we update the stream title to Dr. Junior?
I'm certainly fine with that. I'll send you a picture of it tomorrow.
Oh, please do. That's amazing. We'll tweet that one out just for fun.
Yeah.
So, but yeah, congratulations again. For those of you that don't know, those of us that are tenure track, like that is the major hurdle for our career. And we are stressed out and suffering for 5 or 6 years, trying to get papers, trying to get results, trying to get money, trying to get the community to accept us. And then we get this anticlimactic letter that says you're accepted. And that's what tenure is like.
It's like, okay, I'm going to wake up and write more grants tomorrow. So that part was kind of, oh, look at this, I can climb. Oh, I'm falling down.
All right, Bronco has a question. What would industrial pharmacology look like? Is that trying to synthesize new drugs?
What would industrial pharmacology look like in terms of what?
If you didn't get tenure, you said you'd do it in industrial pharmacology, or you'd go to industry, get paid more, work less. What would that job actually look like? Yeah,
so here's how I view it. Neither one is the right or the wrong choice. It really depends on you, what you want out of life, and what interests you. Okay, how the hell am I supposed to do this? One of the beautiful things about being in academia, in my opinion.
Well, before going into comparing the two, what does an industry pharmacologist look like? Like, as far as you know, what is that?
Certainly. So, it depends a little bit. Within pharmacology, there's a bunch of specialties. So some people are experts in what's called target validation or drug discovery. So depending on exactly what your expertise is, you might be doing different things. So some people are responsible. How do I get over there? There. I had it. Okay. Some people, this one's a lot harder than the other two. So, yeah, hear me starting sentences 5 times over here. Okay, so some people are experts, and their job is basically to identify targets, proteins. Oh my God, this is killing me. Can I stand on this thing? That somebody should develop a drug against in order to treat or cure a disease. There are other people who are experts in designing a way to identify a chemical, millions, that actually binds to and hits that particular protein or target or whatever it is that they're trying to hit.
Oh, so close.
I know. And then there's other people whose job is to take that lead, that chemical that has the activity that you desire, and then make it so that if you put it into, say, a pill, the body will absorb it and distribute it to, oh my God, and then this one moved. Will distribute it.
Oh no.
It's fine.
Oh, this is what grad school's like. Anyone that's interested in grad school, this perfectly describes grad school. You slowly make progress.
My advisor telling me that I did it wrong and to do it again? Right?
It's me. You're disappointing the audience. Work harder.
Okay. Got it. Anyway, so then there's somebody called a medicinal chemist, and their job is to make a chemical that does something, something useful into something that will behave like a drug. Meaning that, again, it'll get into the appropriate parts of the body to do whatever it's supposed to do, and that it won't fry your liver or your kidneys or other important organs like the brain. That last one's maybe debatable. And then there are other people whose job is to test those compounds in animal models to see if they can provide some preliminary evidence that the chemical does what it's supposed to do and isn't going to immediately kill a person if they put it into it. So, all drugs at present have to go through 2 animal models before they are allowed to be tested in humans. And then there are people who run clinical trials. So they take chemicals that appear to be drug-like and didn't kill 2 animals and did what they were supposed to do in an animal. And then they give them initially at very low doses to people and try to demonstrate that they are reasonably safe, that they can do a proper clinical trial to make sure it works. So there's a lot of different people that all have to work together in industry in order to start with the target and end up with a drug that can actually inhibit or alter or otherwise change that target for medical purposes. So I did my PhD in the University of Pittsburgh Drug Discovery Institute. So we were the people who would basically identify chemicals from a large collection of chemicals that had a particularly useful biological function, bound to and changed the function of a particular, this is killing me, biomolecule. So if I were to dump out into industry, probably what I would do. The other thing that I would probably, you suck. Unfortunately, you is me, so I suck.
So starting salary on a position like that is probably 6 figures, what, 100, 120,000?
Depending on where you are and what it's far more lucrative than what you or I are. So the trade-off, though, is you don't really have any creativity, right? You don't control what you target. You don't control, any of the intellectual property, the patents result. They all go to the company. And at the end of the day, you're doing what the company is paying you to do. The beauty about academia is I can study whatever I want. I can pursue whatever I want as long as I can convince someone that it's worthwhile and that they will give me money to do that. I think I'm losing interest in this game.
Brutal. It's something to do with the directionality of the, like if you press arrows before jump, it drops you off or something.
Yeah. So to me, that part was really appealing. But I certainly have friends who went right into far richer than I am or will ever be. All right, one more attempt here.
You are rich in mentoring and teaching students. It fills your heart with richness.
It does. There's nothing better. I call them my work babies. All my students and grad students are my work babies. I have my home babies and my work babies. But, the truth of the matter is I can do all the and, oh my gosh, I suck. Why do I suck? Just because I'm talking instead of focusing on games.
Yeah, no worries.
Yeah. You have far more of an impact as an educator than anyone ever has as a researcher, save for 1 in a million, probably.
Yep. Yeah.
I've trained a huge number of clinicians that are out there saving people's lives, I haven't saved anybody's life. I've probably done a lot of damage to my life, with alcohol and other things, but knowing that, the things I taught somebody has allowed them to go out and make someone else's life even better, I think is priceless. You don't get to really do that in industry. You could potentially have the joy of bringing a drug to market and knowing that was, What do you think? Should we try Contra 3, The Alien Wars?
All right, switch to Super Nintendo. Do you have your controller connected?
All right. I have the controller connected.
I'm going to just shift off that so we can get this set up. Sorry about the brief pause.
All right. Yeah, I am a novice when it comes to game streaming.
No worries. We're on a screen with just my head here.
Ken has been very patient.
Well, so for those of you in the audience, what our ultimate goal is post-COVID is I have a setup at my home where someone can just come over and we can play video games in proximity. But right now we have to do it remotely because of COVID. So it means that we have a sharing software. We're using Discord to share both Robb's stream and Robb's video gaming. And he has a green screen behind him that allows me to overlay things. And so that's where we get the gameplay. And so I appreciate Robb doing this from a distance because it's non-trivial to set this up. But yeah, eventually we'll do this in person and life will be better. And
that will be lots of fun.
so this is Contra 3 for the Super Nintendo.
The
Alien Wars. And so this is on we moved from 8-bit to 16-bit SNES system. So the quality, audio, everything has stepped up its game. And
you know it.
Robb is going to do his best.
I remember this coming out, and it was like we went from 8 colors to 256 colors or something like that. And it was like earth-shattering. I was like, oh my God, what happened?
It changed our lives. It really did.
Right? Oh my God. Super Mario World?
I'm going to argue we grew up in the best time of technological progression because, like, we saw the birth of the internet. We saw video games take hold. Like
cell phones.
Yep.
I mean, everything.
Yeah. Man, I had a colleague of mine who complained about modern students. So, like, you'd think kids today would just be, like, ingrained in technology, but technology has become so user-friendly that they don't necessarily know how to troubleshoot like we had to. Right, in terms of, not everything ran right away, or you had to flip the disk over, or all sorts of stuff you had to troubleshoot.
Those are very bad times.
Let's take a moment to appreciate Calm Down Bronco's comment. This game looks too good for this channel.
It's mostly the player making it. Yeah.
Yeah, no, it's a skill set. Well, pretty much everything we've done so far has been Nintendo. This is, I think, our first Super Nintendo game. So you are right, Bronco. This is some quality gameplay on Ask a Scientist Gaming.
These things are bad. Yeah.
Yeah, this is 1992 16-bit graphics.
Oh yeah. I remember playing these things on my 13-inch cathode ray tube TV that I bought for like $250.
For like a 20-inch.
Yeah. No, it was the one in my room was 13 inches. I saved up ever to buy that.
Did it have a VCR built in?
It did not.
Uh-oh, stream paused.
It was super ghetto. Oh, those were bad times.
Oh, can you share your game again? Sorry, we dropped for a second there.
Oh, yeah. I just paused. Give me a second here.
Discord only lets us stream for a certain period of time.
Okay, my button says stop streaming, and then I'm going to share again. Go live. We good?
Yeah, I think our connection dropped a little bit here.
All right, we're good to go, though? I don't want anyone to miss this action-packed 16-bit.
All right, go ahead and start.
All right, give me a sec. All right.
There we go. Thank you.
All right. Up.
Oh, thanks, man, that was almost three-dimensional. All right, back to questions. So I have a fun one that's I like to ask as well is if you had unlimited budget and no moral qualms of any kind, what experiment would you like to do? Yeah, that is the appropriate laugh for this question. Mad scientist, let it take over. I mean, I asked the developmental psychologist this, so you're not going to go any further than twin studies. So
experiment. So it would have to be an experiment.
Oh, what else are you proposing?
Oh, well, if I had no moral qualms, I would clone myself over and over and over again so that I could get more stuff done.
Is that an experiment or is that just greedy? What's your testable hypothesis on that one?
It's certainly that I can clone myself. That I can augment my substantially and or have all of my days free to play video.
One
by
version of
creating a
you.
number of bitch Robs that I can command. This is a very strange looking turtle.
Oh, cloning. That, man, you did not go for the betterment of mankind necessarily. You went for the betterment
Well,
of
you said no
Rob.
moral qualms.
No, it's true. I need to narrow the
I felt
focus.
almost
Okay.
obligated to do something.
Yeah. No. Okay. So we'll say for the betterment of Rob, that was the choice. What about the betterment of all of humanity? What experiment would give you answers that you've never seen before? I mean, could you just make
You know what I would do? Honestly, I
Yeah.
would make cheap medicines and give them away.
Yeah.
I mean, we use yeast as a model system in my lab. I could make insulin tomorrow, if I could find somebody to pay for it, and just give it away. There's lots of stuff that's made in bacteria and other organisms that we work with every day, and we could do easily. And it would change, an incredible number of people's lives. And it would be really almost no sweat off my back, so to speak. So, it's not necessarily as intellectually rewarding, but I think it would be far more spiritually rewarding, is maybe the best way to word it.
Yeah, that makes sense. So it wouldn't
Plus-
provide new insights. It would solve problems that currently exist.
Yeah. And I'm sort of I'm a bucket biochemist, so I love purifying proteins and peptides and chemicals and stuff. I still enjoy doing that, and I still try to help out the people in my lab when I have free time by purifying stuff and, to speed up their own experiments. And
That's fun.
So-
How often do you still get your hands dirty in lab?
I try to at least set foot in there every day, a few minutes. The thing that I do nowadays is a lot of reagent development for other people. So for better or for worse, I'm probably the best cloner, like genetic cloner in my lab. So I do a lot of the cloning and recombinant DNA work for people in my lab because it's something I can do in little bits and pieces. And I don't have to be like in the lab all day, every day to make forward progress. So I can do it on top of all my administrative and I can't jump this. What happened?
I think you killed all the bad guys.
Oh, sweet. Ooh,
Oh,
look
there
at
you
this guy.
go.
Oh, you're shooting at me. I don't know why I say that like I'm surprised.
Yeah. Can't we just get along?
I thought this guy was going to be my friend, This is the first alien robot with a cannon on the front and spiky, spinny things that tried to kill me.
Man, what I will say about you choosing Contra is, so I have, several guests lined up for this, and they don't want to do the same games as somebody else, and I think Contra is going to be one of the popular ones. But the other ones are like Mike Tyson Punch-Out and Excite Bike, and yeah, some classics.
Seriously, what's going on here? You're making me dizzy.
Nice.
This is a bad time to have vertigo.
All right, so if you had unlimited budget, you did either clone Robs, generate, chemicals for everyone or drugs for everyone. Is there an experiment you could think of that you could gain some new insight answering a pending question? Like, is there a cryo-EM that would, a $1 billion cryo-EM that would answer something, or?
Yeah. So, one of the things that I worked on as a postdoc, this is something that probably would not earn me a Nobel Prize or anything, but it's something that I am really interested in, is, how really complicated pieces of machinery put together, how they're assembled inside the cell. It's one of these things where if you really think about it, it's incredibly complicated. The inside of a cell is super dense. It's as thick as like Jello, realistically. So, things don't diffuse very far. It's hard for things to find each other in space because there's a whole bunch of gunk in the way. All your lines of sight, for lack of a better term, would be sort of.
Sorry to interrupt. We have important breaking news. Bedanning has redeemed Take a Drink. He spent 500 standard internet points for us to drink alcohol, so we will do so now.
That was the very last of my beer.
Uh-oh.
So if there is to be any more drinking, I will have to go get one at some point.
It's up to you. We have about 36 minutes left. Do you want to do one more?
Doesn't matter to me. I don't know if I'm allowed to stop.
You can take a break. It's up to you.
I'm going to keep playing, and if I have to get more beer, God forbid.
So be it.
I'll do what I have to do. I just jumped into a hole. Anyway, so this thing I study, the Proteasome, I think, example. It's got 70 pieces. And if you do the math, there is like quadrillions of different ways that you can put 70 pieces together, and only one of them works inside the cell. You know what I mean? There's only one way, maybe two or three tops that you can do it inside the cell that will yield you a machine. Mm-hmm. And the cell does it right basically every single time. And we haven't the slightest idea how it chooses the, quote, right way to do that, and how it avoids doing it the wrong way, every time when the odds are stacked, 40 quadrillion or whatever to 1. Mm-hmm. So, from a mathematical perspective, that's something that is absolutely fascinating to me. But I think the average person doesn't care. You know what I mean?
Well, so a follow-up to that, if we knew how that worked, what would we do next? Like, we learn how stuff works, and then we control it for our own gains. So what would that look like?
Yeah, so the place where I think that would be useful from a what's the word I'm looking for? So I always default to a medical perspective. Mm-hmm. Look at this thing. He's why isn't he moving? What are you doing, buddy? Okay. From a medical perspective, there is potentially some utility there. There are a number of human diseases, that are characterized by having insufficient or excessive protein destruction. So probably all of you are familiar with Alzheimer's or Parkinson's disease. These are neurological diseases. The brain's control of either memory or bodily function, muscle function, slowly degrades over time. Quality of life is really bad, and in many cases, that's great. It's lethal.
That's perfect timing on the lethal.
I know, right? This guy's stabbing me a lot. So these diseases, these neurodegenerative diseases, are often caused by the accumulation of damaged or defective proteins that should be destroyed, but for some reason aren't. So the body just fails to recognize them, or they can't be turned over as efficiently as they need to be. And over time, they accumulate, and they basically kill off the neurons in certain parts of your brain, and that leads to the disease. So, as I said, the proteasome is really a protein-destroying, protein recycling machine. Being able to enhance the formation of these, to upregulate, to make more of them, might be able to clear out some. Oh my goodness! Stop stabbing me! It's so very painful. Might be able to repair or suppress the symptoms of these diseases. So, but to do that, we have to understand how these things are put together, what the signals are, and how we can enhance it or make it even more efficient than it already is. On the other hand, a lot of cancers have what's called a high misfolded protein burden, which basically means they're messed up and making proteins, but they're failing to make functional proteins. And those are toxic if they're not removed. So there actually are some drugs on the market right now that inhibit protein destruction. They're called proteasome inhibitors. They prevent... Oh, this is good.
Surprise.
Where do I shoot you? Oh, there we go. Gotta shoot in the open eye.
Man, this is no joke. So this is 3 lives, 3 continues. Man.
Yeah, really. Ugh. Anyway, so in those cases, reducing assembly might be really helpful because it would slow down the cancer. So, again, I think there's potentially some utility there. It's just a bit far off, and the United States has gotten in what I think is a bad habit of only funding research that has an immediate application, and they have sort of suppressed curiosity-driven research, for lack of a better term, even though most of the major advancements in biology and medicine have come from these types of studies where people are saying, I wonder if, or, I just kind of wonder how this works. And they discover something that nobody expected, and it turns out to be incredibly useful. CRISPR is a great example of this. This is a bacterial immunity pathway. Most people are saying, who cares how bacteria defend themselves from viruses and stuff, right? But, it led to something that's probably going to revolutionize medicine. And my suspicion is that there wasn't a whole lot of interest in funding this research in the early days, until people started to say, oh, there is a utility. So there's a lot of people who have interesting things that really deserve study and aren't studying them because it gets poo-pooed because there's not an immediate application,
Yeah, I think that's across the board, not just like medical related research, but I see that too.
It's certainly not just biology.
It's basically, your proposed research needs to be guaranteed to work, which means it's, medium risk, medium reward. And the high risk, high reward is just not supported. And it's frustrating. It's very frustrating.
I agree. And I view, and I understand some of the reasoning, right? Taxpayers' dollars, and we need to make sure that they get the biggest return on their investment, right? People are paying money to, oh great, what is this? Their tax dollars are going to fund this, and you don't want to just flush it down the toilet. But at the same time, I feel like it's a stock portfolio where you have some things that are surefire, but maybe not ground shaking. Oh, I should have been climbing the ceiling here. And then I should be doing it again. Oh, he stopped. And then other things that are risky, but if they work, holy crap, it's a game changer, right?
Yeah.
So that's my personal feeling. That doesn't seem to be the mindset of most of the major funding sources for researchers in the United States nowadays, which is kind of a bummer. You have people like Sarah Palin say, ooh, why study fruit flies? Who cares about fruit flies? And the reason is 70% of their genome is virtually identical to ours. And guess what? You can do a lot of things, but they won't let you test it on a human, right? So it's a great way to learn a lot of stuff. So there's an underappreciation, in my opinion, for basic research at the moment that I hope will change.
Yeah, and for those of you not familiar with the process, basically we submit a proposal. That proposal says we want to do this research, and then we'll get that research funded or not funded. And if it gets funded, we pursue that research. But also we use that funds to explore a little bit beyond what we proposed. And so that's where we can squeeze in some exploratory research. But, if I won the lottery and got $1 billion tomorrow, I would basically continue my job, but do so with the flexibility to pursue what I want, right? Because there's stuff that I want to do that I don't have money to do because it's just not going to get funded.
You can't get somebody to fund it. Yep.
Yeah, and it could be good, it could be nothing, but I don't know until.
But if you don't try, you never know, right?
Exactly. So yeah.
And again, it's one of those things where many times it's a game changer.
Mm-hmm. And sometimes it's nothing, but we got to try. But
yeah.
End of our rant. This is frustrated assistant/associate.
Yeah, this
professors.
Is angry scientist talk. Because we ask an angry scientist, or
we apply for a lot of grants, and our proposals are fundable. They are interesting, but you can't fund all of them. So how do you pick the ones you do? And those criteria are sometimes not clear to us. So
yeah.
Yeah, interesting job. But enough about our ranting. Anyone that's just joined us, Professor Robb Tomko playing Contra, as well as answering questions about pharmacology, cancer, biology, drug discovery. If you guys have any questions, please drop them in chat now. If you guys want to follow us, we'll have scientists on hopefully at least once a month, probably twice a month, or more than that when we get back to in-person Ask a Scientist. But yeah, feel free to drop your questions in chat right now.
Yeah, I mean, I'm also happy to answer questions about what it's like to be a scientist, what is the lab like,
Or what is
how
med
do I get
school
into
like?
graduate school, what do I do if I want to be a doctor, any of these things I can help.
So
give my 2 cents on it.
You teach med school classes, right?
I teach pharmacology. I teach some pharmacology. A big chunk of my teaching at the moment is to physician assistant students. Physician assistants are basically, they should probably be called physician replacements because they get a lot. No, I mean, really, they do a lot of the exact same work as proper physicians, but for less of the cred, for lack of a better term. So the plus side, though, is that they have greatly increased flexibility and a faster curriculum or coursework than you do for medical school. So they generally don't do resident like a medical student would. So they get into the clinic much faster. And they tend to have I would say, a bit more career flexibility and life balance than a lot of the medical doctors that I know. So, in some ways, I think it's really quite appealing. But the truth is, as far as pharmacology goes, at least, they get the same curriculum I deliver to the med students. So, they're certainly well-trained clinicians.
So that kind of leads...
on top of that, I also mentor graduate students who are learning basic scientific... Oh, what's this? What do you want? What's it? Oh, he's got a sword. I hear have this super advanced gun, and this guy is stabbing me
and destroying you with the sword. A follow-up on your recent point. So there's a lot of machine learning AI algorithms that are starting to do things like radiology and starting to detect cancer in MRIs and X-rays and things like that. But there are some machine learning algorithms that can essentially diagnose patients, right? Without doctors, they can, ask you the right series of questions. I mean, how does that change the pharmacist/medical discipline in the next 20 years when computers are better at diagnosing than people?
Than people are.
Yeah. I mean, this is outside your domain of expertise, but you might have more insights than I do.
I know a little bit. My feeling is it's going to be... So the thing with machine learning and a lot of these, they're only as good as the information put in. Mm-hmm. So the computer will not always know when to look at a certain part of the body or when to suspect something because it's dealing with a very limited number, right? Whereas at least currently, people still have a creativity that can't really be matched. What am I supposed to be shooting at here? Any idea?
I have no idea, and this looks impossible.
That is... Yeah. I'll accept that. So that creativity... Okay, well, something's happening here. That creativity still can't really be computer, and I think, especially with unorthodox cases, medical cases that are not routine, where things are unusual, it's going to be a very long time, if ever, before we can reliably use AI to them. So I think for, tonsillitis and common colds and garbage that every, family medicine doctor sees every day, yes, you can probably get a computer to do it with really high accuracy. For some of these other things, I think it's going to be a really long... Oh my goodness, come on.
Yeah, this is non-trivial. All right, so it'll be time before we get replaced, which is nice to hear. All right. We have 2 undergrad research questions. So Danny wants to know, have my first interview with a lab PI for a post-bac research position. Post-bac, for those of you who don't know, is post-bachelor's. You go back and take pre-med classes, basically. And so one of the opportunities you can do is research in a lab. But Danning's question is, what is some advice you have? What do you prioritize when selecting applicants for your lab? Any memorable interview/applicant stories?
Where to begin? So
sounds like a good start. Awesome.
There we go. So I cannot tell you how many times pre-med students have come into my office for an interview, and when I ask them why they're interested in the lab, they say they need it on their CV to get into medical school. Which basically, I have no interest in this person. I've never met whether or not they get into medical school. So that's very bad sales. And for someone who is in principle, I feel like that's the stupidest answer you can give somebody. So those are certainly memorable. I can see, I think in my head, the vast majority of people who have said that to me. The things that I actually look for, so this is my view, and I speak on behalf of everyone for things that are slightly different. But when I am interviewing an undergraduate, I have the understanding that this is going to be probably their first real entry point into, okay, I can't go down there, into scientific research. So I don't expect them to, for lack of a better term, know anything or be able to do anything. I view them more of an investment. I look for people who are young and enthusiastic, so that they are going to want to come in and learn things and get stuff done, and they're young enough academically that they are going to stick around for a few years. So the effort that I put into training them is eventually going to yield some dividends to me in terms of them being able to do research that's trustworthy and doesn't require me standing over their shoulder to help them the entire time. And so the things that I'm really looking for when I interview people is really, this is sad to say, but it's usually nothing on their CV. It's really, when I'm explaining things to them, are they interested? Are they asking questions back? Are they processing it as I'm telling them about it? And I think that's really one of the best predictors of whether or not someone will be productive and have a good time. This is really complicated. Whether they're going to be productive. I don't even know if I'm supposed to be shooting this thing. All right. If I was super drunk, this would be really painful.
Yeah, it's starting to lag. It's too much stuff on the screen. No, I mean, going back to your point, there's research that basically supports that success above a certain intelligence threshold, your success in terms of research has nothing to do with intelligence. It's all time spent, work ethic, make mistakes, don't make them twice. Like, enthusiasm and work ethic are huge. I'll take a motivated English major over a chemistry major any day. Like, give me somebody that's going to get shit done and they're going to be productive in lab no matter what.
Yeah. I mean, I'm not claiming to be dumb, but I'm not the... But, a lot of where I've gotten has been because I'm willing to grit my teeth and bash at something when, the other 10 people who were thinking about it weren't. You still have to have enough intelligence to do... Can I jump? No, I can lay down. That would have been useful to know when I was stuck in those spinny things. So I mean, that's gotten me further in life, unfortunately, for better or for worse. I was fortunate enough to postdoc at Yale, one of the most prestigious institutions in the world. And, there were people there that were definitely smarter than me, and they would sit on their computers and shoe shop during the day when they were supposed to be doing experiments instead of experimenting. And I got further than they did, So there's a lot to be said for grit and enthusiasm, in my opinion.
With that said,
I will take that over rote intelligence, I think, probably almost any day.
With that said, there is a healthy dose of just flat out luck to success in science. Like timing in what you're studying, timing in what you're working on, seeing something no one's ever seen. It's underappreciated how people just get lucky sometimes.
Yes, that is absolutely true. Having said that, you can not get particularly lucky and still do just fine,
Yep.
but you may not win your Nobel if
Yeah.
that's what you're into.
So like in my group, we specifically will pursue a few different projects per student. One is going to be high risk, high reward. If it works, it's amazing. Other ones are going to be guaranteed results, like things that no matter what you do, it is publishable because knowledge will be gained. Change some systematic variable and somebody wants to know how that works. But all of it
Yep.
comes down to putting in the time and just grinding, like getting stuff done,
Yep.
spending the hours.
It's like getting that XP, man.
Yeah, exactly. No,
Gotta do it.
it's the World of Warcraft of science. I thought about it that at one point, like figuring out some kind of reward system in lab where you could like watch your numbers go up, because there's something very visceral about gaining arbitrary internet points and seeing your skills increase and like having your skills develop. But I never
Yeah.
expanded beyond that.
Yeah, so I think that's important. The one thing that I do tend to look at on resumes is not necessarily exactly what somebody has done, but how many things they have. This is again something that I see very commonly in pre-med students that want to do research. They have a laundry list of activities and things, and they all lasted 3 months, and that was it. And it raises a concern. Unfortunately, that's par for the course, I think, to get into medical school. Sometimes you have to check X number of boxes.
Mm-hmm.
But, it makes me wonder whether you have the commitment to really bash at something for a long period of time. So I don't exclude people per se because they don't have that. But if I find somebody who, say, worked at the same job all through high school, or was involved in the same club for many years, and they may not have done a whole lot of other stuff, but it was clear that they made a big impact in that one thing. I also find that attractive.
Danning, in terms of the interview, I would emphasize that you're really there, you want to learn, you want to contribute, and maybe mention, I want to do everything I can to get on a paper. Like, say that line. Those are magic words for a professor that wants to publish.
Yes.
Like that means
that is fair.
You want to put your skin in the game and you want to earn a publication. Because make no mistake, that publication means everything for grad school. I don't know about med school, but grad school, get on a paper.
Less so in medical school, but depending on what you want to specialize and so on, it can matter. And some people do MD PhDs where they a few years in between their didactic coursework in medicine and their clinical work, basically to get a PhD, and obviously they need a paper to graduate or whatever as well.
Yeah, so, their MD PhD, that's for experimenting on humans, right? That's next level research, right?
So I need to be careful what I say. What I have found is that they generally are given projects that are low risk and likely to succeed in an effort to get them back into medical school very quickly.
Oh, that's for the graduate portion of it. Yeah, that makes sense.
Yeah. That's not to say that it's easy. It's certainly not to do medical school and a PhD. It's like doing two really hard things when you could really just do one, right? But, in fairness to the people who grind for 5-6 years.
We just game overed.
I know.
I don't know how that happened. You died enough on this level type. It's crazy.
I can't figure out what I'm supposed to be doing.
I think it's blow up those things like that thing. You want to blow up all of those. Is there
a way to? I think a map. There's a map. I'm learning so much.
Oh man.
Okay, this changes everything.
So we haven't made emotes yet, but one of the emotes I want to make is a person's head with a light bulb, both scientifically and in gameplay. We're gonna have light bulb moments like that. All right, Danning, hopefully we answered your question. Calm Down Bronco, do either of you have undergrads working in your lab?
Yes. I have, I think 5 undergrads in my lab right now. and I just had one leave in December. She graduated and she's interviewing at schools right now. I love having undergrads in the lab because they help to keep me young. This is how I learn about things like TikTok and Azalea Banks boiling her cat, apparently, if you guys heard about this morning. I don't really find out about anything anymore unless an undergrad. So, all right, I'm really losing focus.
Oh, so you get deaths if you fall off the edge. Don't fall off the edge. I think you're invincible.
Roger that.
I think our Game Genie codes will cover you as long as you don't fall off the edge. But all right,
I'm
so we
hoping
learned.
no one's will actually at this point because I'm not doing very well.
We're learning that undergrads are good for keeping up on pop culture. I'm surprised. So chemistry, they've limited us to 2 undergrads during COVID, and that's just occupancy restrictions and things like that. So
we were at 50% occupancy, but we were allowed to do shifts.
I see.
So my lab, we had some people, where am I going? That'll work. We had some people would come in super early. So the old people, basically like myself, would go in at like 5:30, 6 a.m. in the morning. Where am I? Okay. And work till like 2. And then, grad students and undergrads, people who can stay up past 9 o'clock at night on a normal Tuesday, would come in and work the second shift, and we could stay below occupancy rules that way pretty easily. Mm-hmm. So we were somewhat fortunate in that we were able to sort of keep everybody moving and rocking and okay.
Oh, question directed at me. Do you currently accept undergrads, Ken? I wish I could, but I'm at that occupancy number.
I'm the guest. No, I'm kidding.
No, I'm recruiting students for lab. No, I would love to, but I'm at that 2-person occupancy number, so I can't until they graduate this summer. So, yeah, it's brutal. But Rob is looking if you need students.
All right, I got another one. What's left? Of course, the other one's like 80 miles away. All right. Gonna limber up. I'm feeling good.
You got this? So we have
I think I finally got in the hang. The map is... I still can't avoid falling off cliff.
If my interview fails, Dr. Tomko, I will send my CV.
Awesome.
So you potentially have a candidate.
Life is good.
All right.
I'm always on the lookout for motivated people.
So we have 9 minutes till closing time of Ask a Scientist Gaming. We should do a quick poll.
So help me, I will
see
finish this level.
If he's going to beat this game. We don't have a lot of people left in chat, but we're going to start a prediction if we can.
That's all right. We have the important ones, right? What do I get? Ooh, this one looks fun. Where am I going? All right, so across this way. Oh, this is so difficult to navigate. What are these things? Are these like sand or?
I would avoid those bridges. You've died repeatedly on those, and I don't know why.
Which ones?
Oh, you made it on that one. I don't know. They fall up. They like.
Oh,
fall
the bridges. They
sometimes.
they break. They break. Am I stuck over here now, or can I get somewhere else?
All right, so
okay.
we've popped up a prediction. Do you think Rob is going to beat this level in the next 8 minutes?
That's not looking promising.
Use your standard internet units. Risk them. Do you have faith in Rob going to beat this, or is he?
I'm going to go slow, because now I feel like if I don't, I'm a failure.
I mean, you have a time limit too. We're doing 8 minutes. Are you going to beat this before the 11 o'clock time window?
It's happening. Look at me go. Look at me go.
You got this. I'm not going to tell you whether the channel has faith in you or not.
They don't. It's fine. I know.
No, this is what makes it fun.
I know. I like to think there's someone out there. But
I like that you're being meticulous now. Like you're earning this one.
Oh, come on! I did not make it. All right. What do I have? 7 minutes? I can do it. Oh, no, it continues.
Oh, no. The prediction is over with 100% voting no. Everyone wins.
I love it. Except
Rob,
me, yeah.
not beating this.
All right.
Oh,
So
congratulations.
Let's explore my options here. Player. Oh, see, I could have go. Oh, nice. And then, can I make this easier? Hard. Let's go hard and see what we get.
What were you playing on?
Normal. It gets worse.
We should have gone easy, even with Game Genius.
Yeah, maybe.
Oh, the results are no. Congratulations to everyone that predicted that correctly. F in chat for Rob for failing miserably at that challenge.
Thank you. I'd like to thank the Academy for believing in me.
Oh, that was amazing.
And that guy for running me over. I like this first board. See, I'm old school enough that I still really run to the die. This overhead stuff is killing me.
Yeah, it's very weird, isn't it? What's with this pseudo three-dimensional stuff?
Yeah. And is that a dog eating out of the trash?
Because everyone voted no, there was no.
See what he's eating.
There was no net points. That's amazing.
Okay, there's definitely a lot more guys.
All right. Anyone joining us at the last minutes of the stream, we're going to go till 11 o'clock. Joining me today is Dr. Robb Tomko, pharmacologist, Contra player. Is happy to answer any last-minute questions you have about drug discovery, cancer, Contra. He will address anything you have. Med
I think
school.
if we've learned anything here tonight is that I don't know.
I mean, you did well on Contra and Super Contra.
That's true.
I think if you committed on Contra, you could do a speed run.
Yeah, I mean, I was in 19 minutes. I got to look at that chart before about what the top 250 times or whatever. I mean, I'm not so far off from top 100, I would think.
Yeah, so if you go, you have to beat it legit, right? No Game Genie codes. But if you do it in under 20,
that's right.
If you do it in under 23 minutes, 28 seconds, you are 235th for speedrun ranking.
Yeah.
So practice
So can
up.
you use the Konami code?
Nope. Has to be legit 3 lives. I mean, most likely you'll want to have deathless or only a couple deaths for that time window, but yeah.
Certainly. So you do realize this is all I'm going to do.
Oh, no, absolutely. And I look forward to having you on again. And we will say Contra speedruns with Dr. Robb Tomko, who answers pharmacology while speedrunning.
We can call it Rob on speed.
We'll call it Dr. Junior on speed.
Dr. Junior on speed. Perfect.
All right, last minute question. Aside from the high felt by them, what makes painkillers so much more addicting to people than non-painkiller drugs?
What makes painkillers so much more addictive than non?
Yep.
Okay. So the short version is that they stimulate what's called the dopamine center in your brain. So dopamine is basically your brain's reward system. It's how you learn food good, pain bad, all these types of things that when you were a cave person, before you evolved, were really, really important to keep you alive, right? The problem is we're exposed to a whole bunch of stuff now that does not exist in nature, but can impact that pleasure center or that reward center. And our body always doesn't really know how to interpret it, but it knows that anything that dings that reward center is something we should try to keep doing. And this is basically why people become addicted to drugs, is because it triggers that dopamine center, which is this ancient, survival system in your body that says, good, keep doing this, keep trying to do this, keep getting this, whatever. And it's nowadays, it's disconnected from, the actual survival benefit. So painkillers and some other drugs are really, really good at stimulating this dopamine center in your brain. And that gives you a very strong positive reward, for lack of a better term, when you take the drug, and it makes you want to take it again and again. So other drugs that can kill pain, so aspirin, ibuprofen, naproxen are all examples of non-opioid painkillers. They kill the pain by acting on the molecules that trigger pain in your body, but they don't trigger that reward system in your brain, is the short version. So basically, we're fooling evolution with opioids and some of these other things. In fact, every time you surf Facebook or whatever, and you like something or whatever, or somebody likes your post, you get a little burst of dopamine too, which is why you see all these people addicted to their phones and social media and all this kind of stuff. It's the same idea.
Speaking of which, make sure to push that follow button right now.
Yes.
Follow Ask a Scientist Gaming. Give me my dopamine.
You want dopamine. It's awesome.
You'll get notifications when we're going to go live. Follow us.
Yeah.
All right.
And the best part is you can't be arrested for clicking the follow button like
That's
you can
true.
for using opioid.
Very
There's
true.
no risk of death as far as I'm aware. Can you confirm that?
Unless you get raided and swatted. Anyway, different problem entirely.
Really. So, yeah, I mean, that's a really big problem nowadays, trying to find alternatives, alternative drugs that are able to work the same way to suppress pain that opioids do, but without triggering that dopamine center in the brain. And that's one of those things that's easier said than done. It's kind of like trying to disconnect two things that both have to be connected to function. So hopefully we'll make some headway there. We being people who work in that area, I don't. But, again, it's an area of active research where people are trying to find some better alternatives that are less addictive. So that, and the government is finally, I think, cracking down illegal opioid prescription writing. Most clinicians are now aware of the substantial problem that the opioid crisis represents, and they are minimizing the prescription of opiates as well as painkillers, and they're trying to defer patients to non-addictive painkillers as a first approach whenever possible, and only using them as a last resort. And the interesting thing for all of you is that the type of pain that you are experiencing actually dictates the best drug to use. There are some types of pain where opioids are practically functionless, and something as simple as aspirin or ibuprofen will work great, and vice versa. So there's a lot of different types of pain, even about it that way most of it. How am I supposed to know where that bridge is going to stay intact?
So we're over time limit, but I'm curious, what different types of pain? That's just different receptors or different neural pathways? How does that?
Yeah, so pain is sort of a collection of sensations that are interpreted by the brain as bad, right? So you can imagine that being feels very different than being cut, right? So the reason is that the receptors that are signaling that are responding to different biological cues. In the case of being cut, it might be tissue damage. It's detecting that some cells have burst open and they've released a chemical that shouldn't normally be on the outside of the cell. With a burn, it could be stimulation of temperature sensor receptors on the part of the body that's being burnt, sending signal. And these get sent to different parts of the brain and integrated differently by the brain. So depending on the source of the pain, one drug may work better than others. So, ibuprofen works really well for inflammatory pain because it suppresses the source, which is inflammation. So, when people get surgery, oh, there he is. Why wouldn't he pop out? This is like whack-a-mole. When people get surgery, most of the pain is from being cut open, right? So the truth is, things like anti-inflammatories are often more effective than opiates actually are for pain suppression in some cases. All right, I don't know why this guy isn't opening up, but I'm pretty sure I had to kill him. I had to kill him, right? Yeah.
Either way, we are past 11
Out of
o'clock
time.
and we are old. Not necessarily out of time, but I think this is a good time to call it quits.
Certainly.
Thank you, everyone, for stopping by. It's been awesome. Rob,
I had
any
a lot of
parting
fun.
words?
Thanks for the gifted sub. Awesome. Calm down, Bronco. I had a lot of fun answering questions. A lot of good questions in there.
And playing
Come back
Contras, getting 3 hours of gaming time without kids or family.
I know.
We love our
Probably
families.
the longest time I haven't gotten up in a long time.
We love our families, but it's awesome to sit down and game for a long period of time like back in the day. So
It's important to have me time every now and then, right?
Thank you, everyone, for swinging by. Follow us to get notifications when we go live. Follow our YouTube. This entire stream will be up on our YouTube channel tomorrow night after 11 o'clock. And then, follow us on Twitter. You will get notifications about Next Scientist. We typically do right now, maybe every 2 weeks to a month, I'll have a new scientist on. So thanks for swinging by. We look forward to doing it again at some point. So... Stewart Valley next, super chill farming game, less stressful than Contra. I agree. We should do something chill. Thank you, Dr. Junior. And with that, we will
Valley
close.
Sweet.
Thank you, everyone. We'll see you guys next time.
Awesome. Dr.